Tem uma hora de pergunta, alguma coisa assim.
Good morning. Before the presentation, I would like to remember that we changed the time of the coffee break. We will have the first presentation, a coffee break, and then the other two. So for me, it's really an honor to welcome these three outstanding researchers, with whom I have the pleasure to collaborate. Their contribution to the field of epigenetics and oncology are remarkable. I hope that you enjoy this opportunity to learn more about their work and also to interact with them. I would like to briefly introduce our first speaker, Dr. Frank Slack. Dr. Frank Slack is a professor of the Department of Pathology and the director of the RNA Medicine Institute at Beth Israel Deaconess Medical Center at Harvard Medical School in Boston. He received his Bachelor of Science from the University of Cape Town in South Africa before completing his PhD in molecular biology at Tufts University School of Medicine. He began to work on microRNAs as a postdoctoral fellow in Gary Hufkens' laboratory at Harvard Medical School, where he co-discovered the second known microRNA, LET7. His group has worked on the discovery of novel small RNAs in development, cancer, aging, and diabetes, as well as identifying new SNPs in the non-coding portions of the genome, with special interest in identifying the next generation of actionable targets in cancer. I invite to the stage Dr. Frank Slack. Thank you for being here.
Thank you.
Okay, thank you.
Thanks very much, Miriam. Thanks everybody for attending. Thanks for the invitation and the nice introduction. I believe I can move my slides forward like this, with a point like this. Okay. So it's great to be here. And as Miriam was saying, we spent the last 20 years or so first as a basic science lab and now a translational science lab. We're trying to take new discoveries in non-coding RNAs and translate them into new medical applications. Today, I'll spend most of my time talking about our efforts to generate new therapeutic targets from non-coding RNA genes. But most of the success in this space in terms of actual translation and medical applications has come in the diagnostics area. I won't talk much about that today. And I say in my title, towards microRNA-based cancer therapy, because unfortunately, I cannot say that we are there yet. This is going to be a talk that I hope will give you a sense of the journey, how we've taken some of these discoveries, and through, I would say, a little bit of trial and error to a certain extent, have moved into clinical trials. And at this point, unfortunately, there's not an approved drug yet, but the hope is that in the next few years, there will be something approved in this area. So as I go through this talk, I won't be able to mention all of the collaborators that I've had over the past 20 years, but I've listed some of my collaborating labs here, including Miriam's group, because without them, I don't think we would have made the advances that we've made. I'm originally a developmental biologist. I'm a developmental geneticist, and I'm not an oncologist. I'm not an MD. I'm not a biomedical engineer. I'm not a bioinformatician. So these labs have helped us immensely along the way. And then I just want to mention also that I'm a founder of some companies in this area. So for sort of full disclosure, I'm going to be talking about some work that I did with this company, Miragen, in one part of the talk today. But I have no financial interest in this company. So I was on the advisory board for the company, but I have no stock in the company. All right. Let's see, does this work? No. Does this work? Yes, okay. So I'm going to begin just by giving you a little bit of background into why I find this area so exciting. Of course, it's been clear for the last 20 years or so since Science magazine listed non-coding RNAs as their breakthrough of the year back in, I think this was like maybe 2002, something along those lines. And then, of course, it hasn't escaped the notice of the biotech and pharma world that these now are a new paradigm for diagnostics and therapeutics as well. We'll talk mostly about this particular area over here today. Now, I've been in this field for long enough to see the ups and downs in the sort of adoption of this technology into the clinical space, and I would say that at the moment we're on a definite up. RNA medicine is undergoing a renaissance right now. Not only just the number of pharmaceutical companies, biotech companies that are working in the space, but also the fact that over the last 5 years or so, we've had a number of really successful clinical drugs that have entered the market. In particular, the RNAi drugs, the siRNA drugs from a company called Alnylam. There may be 5 or 6 now that are approved for treating a variety of different rare diseases. There are antisense oligonucleotides that are now on the market to treat a variety of neurological disorders. This is one that treats spinal muscular atrophy. And then, of course, many of us were the benefit of getting a messenger RNA vaccine during the COVID time. And now we'll continue to get mRNA vaccines for other infectious disease and maybe even for cancer going forward in the future. I also want to say that RNA medicine is also getting highly personal because of the fact that these are programmable drugs. They're basically drugs that are based on sequence. They can be designed to target any nucleic acid sequence that you want. You can target a particular patient's genetic makeup with these kinds of drugs. And this is the first example of it. This is a paper that came out in the New England Journal of Medicine, where they used an antisense oligonucleotide against a mutation in a child that had a neurological disorder called Batten's disease. and we're able to treat this child for at least a few years before the patient ultimately passed away. But this was a case where the child had a neurological disease without any known cause. The researchers, as described in this paper, did a whole genome sequence on this patient and identified a mutation in an intron that affected a splicing of one particular gene that for which there was no drug. So they designed an oligonucleotide that altered the splicing around that mutation. And like I said earlier, this patient received the drug and had fewer symptoms, but ultimately passed away. So it's not like these drugs are perfect yet, but they're getting to the point of being effective and being highly personalized. All right, so why is this such a, these are early successes, why are they so potentially impactful for the future? Well, the reason why they're impactful is that we have a lot of RNA that's made within our cells. We have a lot more RNA genes than we have protein coding genes. So just, as many of you know, the Human Genome Project, when it was published in the early 2000s, revealed that humans have only about 20,000 protein coding genes. And for some of the people with gray hair in the audience, you'll remember we thought that we would have 100,000 genes because we were so complex and so special. But we actually have less genes. We have half the number of genes as a tomato plant. All right, so we're not that special in terms of our protein coding genes. It turns out we have a lot more non-coding RNA genes. So for example, just if you look at this pie chart, these are our protein coding genes. We probably have 50,000, maybe the number is even more like 100,000 now, as this list includes new discoveries. But maybe 100,000 long non-coding RNAs that look a lot like a messenger RNA, but aren't translated into protein. Then, of course, you've got thousands of short RNAs, like the microRNAs that we'll talk about today. If you look at the number of variants of the microRNAs, well, we may only encode 2,500 in our genome. There are a lot of different variants that have extra nucleotides in the front and the back, some changes in the middle. There are about 250,000 of these isomers in our cells. About 100,000 Pee-wee-associated RNAs or piRNAs that are made in our testes and ovaries that are used to silence transposons. I've already mentioned the long non-coding RNAs, many pseudogenes that sort of fall in this long non-coding RNA class. Many genes are backspliced, so they have a forward linear spliced version of the RNA, but also a backspliced version that makes a circle. So we have 100,000 circular RNAs. And we're also appreciating now that many RNAs sort of moonlight with other roles. So many of our RNAs get processed into smaller and smaller pieces. And for example, some of the tRNAs that we know and love for their role in translation, they get cleaved into smaller fragments called tRNA fragments that also have functions in our cells. So if you add to that the fact that many RNAs get modified at a variety of positions with about 150 different modifications, what I hope you'll take away from this slide is that there's an immense amount of complexity in our cells. But what I also want you to take away from this slide is that this provides an immense amount of opportunity. So new opportunities to understand disease and to target disease. If you can think about just trying to target these protein coding genes with the current drugs that are in the market right now, there are only about 3,000 protein coding genes that are amenable to small molecule inhibition. For example, our kinases, our methylases, G protein receptors, that kind of thing. So 3,000 out of 20,000 is not a lot, but when you think you might be able to add another 100,000 RNA targets to that list, I think you can see the opportunity is going to be great. But where we're at right now is we're at the sort of, oh my goodness, there's so much here to do. How are we going to understand what all of these things do? So one of the things that my lab tries to do, and one of the things that our institute tries to do, is bring more people into the field to try and study and understand these genes with the hope that we can find the ones that are important for disease and target them with these sort of RNA medicine approaches that I mentioned today. So the genes that we'll talk about today are coded, are actually our genes that code for microRNAs. These are very short RNAs. They're only about 20 to 22 nucleotides in length in their mature form. I don't want to go through this biogenesis pathway in much detail today, just to except to bring up a few points that are relevant for cancer today, for most of the talk. The first thing I want to talk to you about is that, in fact, microRNAs are generated from our own genes. They get transcribed from our genes. And this is an important point, because in the early days of this sort of discovery of microRNAs, it wasn't clear that where they came from. But now we know they're transcribed, they're endogenous, they're made from our own genome. They don't come from an infectious agent or from the environment. And so, because they come from our own genes, in many diseases they get mutated, they get deleted, they get amplified, they get translocated. Anything that you can imagine that happens to a protein coding gene in a disease like cancer can happen to a microRNA gene. And so we and others have been trying to identify the microRNA genes that are mutated, amplified, deleted, upregulated in disease, and I'll tell you about our work today in cancer. The other thing I want to point out is that there are a number of different enzymes that are required for the processing of these RNAs to their mature form. And many of these enzymes are made from genes that we now know also are mutated or deleted or upregulated in cancer. And then the last point I want to make is that the microRNA is essentially a single-stranded small RNA that enters the RNA-induced silencing complex and guides this protein complex to complementary sequences in the genes that are going to be silenced by this complex. And why that's interesting for cancer is that in almost every case that we've looked at, the most important cancer genes, your favorite cancer genes, are all going to be regulated by microRNAs. I mean, I could put money on it that if you have an interesting cancer gene, in its 3' untranslated region will be microRNA binding sites, and you will be able to identify microRNAs that regulate that gene. In fact, we now know that microRNAs fit into almost all of the most important cancer pathways. So why that's interesting is because we think that we can use microRNAs now to control the expression of these cancer genes, and we're going to use that as a potential way to design therapeutics for cancer. I don't want to go into this in too much detail, but we'll just point out one thing, and that is that in many cases, microRNAs are made more abundantly in certain tissues than other tissues, and so you can, you almost always have to profile the tissues independently to make certain that you've identified the microRNAs that are most important for that particular tissue. This is a very early experiment we did way back when, showing an in situ hybridization of a mouse embryo, looking with the LET7, looking to see where the LET7 microRNA was expressed. And we can see it's expressed in parts of the brain, parts of the heart, and parts of the lung. And that became sort of relevant to us as we identified this particular microRNA is important for lung cancer. But I won't talk about that particular experiment today. What I'm going to tell you about is some examples of how we can take this new information and potentially design new therapies. So what are the opportunities for RNA-based therapeutics? Well, there are a number of them. I'm going to give you some examples from this first and second class today. In the first class, we can think of the RNA as a targeted therapeutic. So we can actually deliver an RNA as a drug. This is most apparent in, for example, the siRNA drugs, the RNAi drugs that are really available to patients with certain liver diseases. They get an infusion of an RNA that goes to the liver and targets a particular gene in the liver. But in our case, we're looking at, for example, tumor suppressor RNAs. Maybe they get deleted in a cancer type. We want to deliver those RNAs as a potential tumor suppressor therapeutic. In the second case, we want to think about the RNA as a therapeutic target. So maybe the RNA is upregulated or amplified in your disease, in which case we can think that the RNA could be drugged. Can we design an oligonucleotide that could inhibit the activity or the expression of that particular RNA? And so in this case, we might think about inhibiting an oncogenic RNA. And I'll give you some examples of both of these cases today. And then lastly, you can think about trying to target, using a more traditional approach with small molecules, trying to target the enzymes that make these RNAs. That's something I won't really talk about today, but there are companies that work in that space as well. All right, so I'm going to just give you one example of how I think this is sort of moving its way further and further into the clinic by just pointing out that if you look in the clinical trials database called clinicaltrials.gov that tracks clinical trials all over the world, and just look just for the keyword microRNA, you can see that there are about 1,350 clinical trials in the world right now using microRNAs. Now, most of these clinical trials, as you can see, if you can read this, you don't have to read this, but take my word for it that most of these clinical trials are in diagnostic applications. So they're using a microRNA or a panel of microRNAs to try and diagnose a disease or determine which particular drug might be most appropriate for a patient. But there are about 24 or so clinical trials with microRNAs as therapeutics, where they're delivering a microRNA or they're delivering an antisense molecule to a microRNA. And we'll talk a little bit about some of those examples today. If you come to my course tomorrow, the next day, I'm going to tell you that the number of clinical trials with other classes of RNAs is also on the rise. So there are clinical trials now for long non-coding RNAs, for circular RNAs, for pi RNAs, for tRNAs. The number is just increasing. And I think it's got to do with the fact that there are just so many opportunities here that we can now exploit for these new therapies. All right, so today we'll focus this talk on roles for microRNAs in cancer. One reason I'm doing this is, first of all, this is a disease area that microRNAs sort of first emerged as being potentially useful. But I can tell you that probably most of the clinical trials now that are in microRNA, that use microRNAs as a therapeutic are more in metabolic disease, diseases where you can deliver the RNA quite effectively to the liver or to the kidney. And so the clinical trials have been in, for example, hepatitis or in liver fibrosis or in kidney fibrosis. But the emergence of microRNAs as potential therapeutics came from the cancer space. So let's talk a little bit about, just so briefly, about why we think microRNAs are potentially useful new therapeutic targets in cancer. Well, the first is, I mentioned that they're frequently located in genomic regions that are altered in cancer patients. So back in the year 2002, something like that, cancer oncologists in Carlo Croce's lab started to just examine their patients. They knew that their patients had mutations. They couldn't find where those mutations were because all the protein coding genes looked normal. But when microRNAs were discovered, they finally clicked. Maybe these patients have mutations in the microRNAs, and they started to sequence the microRNAs from these patients and identified a number of cancer-associated mutations within microRNAs. In fact, they published a paper that a few years later showing that about half of human microRNAs are in regions that are known to be amplified, mutated, deleted in cancer patients. We and others had done these sorts of experiments with sort of microRNA microarrays, and we showed that microRNAs were almost always misexpressed in tumor relative to normal adjacent tissue from the same patient. We're going to talk a little bit about this example today with this, in particular, this microRNA called miR-155. You can see miR-155 is highly upregulated in one particular type of lymphoma, but not in another type of lymphoma. I've told you that there are mutations in the microRNA biogenesis machinery genes. The most famous of these is DICER. DICER is the enzyme that cleaves the double-stranded RNA to make two single-stranded RNAs. And there is now a well-known syndrome called DICER1 syndrome, where children inherit a mutant copy of DICER from their parents and then have a loss of the second copy early in life, and they get childhood cancers. We and others have shown that microRNAs regulate oncogenes and tumor suppressors, and we've also shown that they can, by themselves, be tumor suppressors and oncogenes by building mouse models. And I'll show you some of those examples today. So about middle of the 2000s, we started to ask whether there were SNPs and genetic variations found in these non-coding portions of the genome, and we've identified many, including some in microRNA genes, and most of them actually falling within microRNA binding sites within known protein coding genes. So in the early days, we found, for example, SNP within the KRAS 3' UTR that affected binding of a microRNA. We showed a SNP within the BRCA1 3' UTR that affected binding of a microRNA. And I can tell you that in almost every case, these are not alleles that are typically screened for in your favorite cancer panels. So we've been trying to get the field to adopt a more expansive view of mutations. They're not always in the protein coding region. They can sometimes be in the 3' UTR or the 5' UTR of a gene. And then lastly, they're stable in body fluids. This is something that that many of you are already aware of. You work on non-coding RNAs that are found within the blood or within urine or tears or whatever tissue that's your favorite to work on. And so they're becoming useful as diagnostic and prognostic biomarkers. And if you come to my course in a few days, we'll talk a little bit more about this area as well. So the fact that there are now prescribable tests that you can get, maybe not in Brazil, but I hope soon, which measure the levels of microRNAs in your blood and can give you a pretty good assessment of your cancer risk or whether you'll respond to a therapy or not. Okay, so for the first sort of story, I'm going to talk a little bit about how we translated a discovery of a microRNA that was upregulated in a cancer type into a potential therapy. So I mentioned earlier that miR-155 is a microRNA that we and others had shown was highly upregulated in a number of different human lymphomas. And so in order to ask whether miR-155 was in fact causal in the lymphoma or was it just like a passenger or a marker for the lymphoma, we built a mouse model where we took just the miR-155 genome, about 150 nucleotides that expressed the precursor miR-155 behind an inducible promoter, and we inserted that into the ROSA6 locus in mouse. So some of you who do mouse work, you'll understand that what we've done is we've made an inducible mouse where we can add doxycycline and turn the gene on and off at will. What we found, first of all, was that if we express miR-155 in these mice, they get lymphoma, which was about as good a result as we could possibly have imagined because we were hoping that we would be able to model the human disease. So I'm not sure if you can see this, but these mice have about a golf ball-sized lymphoma growing from the axial part of their neck. So this told us that indeed miR-155 could be causal in lymphoma. You just turn on this one gene and the mice get the disease that you think they would get based on the expression pattern in humans. And I should tell you that this mouse expresses miR-155 about 50-fold more than in a normal lymph node. And that's about the same upregulation that we see in human lymphoma patients. So this looks physiologically relevant and in many different areas. We also got a pathologist to blindly diagnose what disease these mice had. And his diagnosis was that they had follicular lymphoma progressing to diffuse large B-cell lymphoma, which was essentially what humans get if they, or at least sort of, if miR-155 is upregulated, many patients will get this diffuse large B-cell lymphoma. But I'll tell you in a minute that it's one particular subclass of diffuse large B-cell lymphoma called the double hit, which is they upregulate MYC and they upregulate BCL2, which are two well-known oncogenes. All right, so the other thing that this experiment allowed us to do was to switch off miR-155. So we could let the animals develop tumors, and then we could switch off the gene. You change the levels of doxycycline in their diet. And remarkably, in every case, the animals went from looking like this to looking like this within just a few days. I mean, these tumors completely regressed. I'm not going to be able to show you some videos because I believe the videos have been deleted, but these mice, just so you know, they get disseminated lymphoma. It metastasizes all over the body, into the brain, into the spinal cord. They eventually become paralyzed and they cannot feed themselves and ultimately die. But within just a few days of getting the gene switched off, there's no evidence of lymphoma at all in these animals. So what that told us was that these tumors were not only required miR-155 to form, but they required miR-155 for their continued survival. So these tumors were addicted to miR-155. And why that's interesting is because what this meant is that maybe we could target miR-155 as a potential therapy for lymphoma. Lymphomas will be addicted to it. You target miR-155 and the tumors would die. And this is the basis for a lot of current cancer therapies. You can think about EGFR tyrosine kinase inhibitors that target a mutant EGFR. The cancers are addicted to EGFR. You target EGFR, the cancers die. And so using this, we started to design antisense oligonucleotide approaches to try and target miR-155. One of those examples is shown here. We have an antisense to miR-155. We used a targeting ligand. I'm not going to go into the details of this, but just point out that it does go very nicely to the lymphomas and also to the cells if they're grown as a xenograft in the flank of a host animal. And we could see the same result, that these animals completely regressed when we treated them with an anti-miR-155. So unfortunately, I believe these are no longer videos, but if they were, oh, they are working. Look at that. No. Okay, so they are working. So this is the animal before injection. It's almost paralyzed. We gave this animal a dose into its tail vein, and now within a few days, it's sort of happily moving around the cage. Now, for those of you who do mouse genetics, you will recognize the fact that it's fairly easy to treat cancer in a mouse, especially if you know what's causing the cancer. And in this case, we know miR-155 is causing the cancer. We treat it with an anti-miR-155 drug. The animals go away. It's much more, the tumors go away. It's much more difficult, obviously, to treat cancer in a human setting because of the variability both within patients, but also within the cells of a human tumor. I should mention that in this particular mouse, when we profile the tumor cells from these mice, they are clonogenic, which means every single B-cell lymphoma cell originated from one individual B-cell back a few months earlier in the mouse. They're clonal. They have the exact same B-cell receptor. The VDJ recombination is identical. So, but obviously that's not the case in human beings. It's much more variation. So we teamed up with a company called Miragen Therapeutics to ask whether we could work with them to design an antisense oligonucleotide against miR-155 that could be used in a clinical trial in human beings. And this is the page about that clinical trial from clinicaltrials.gov. It's basically an antisense oligonucleotide to miR-155 called MRG-106. You can see that MRG-106 is an inhibitor of miR-155 that is a particular chemical structure known as locked nucleic acid. I'm happy to talk to you guys, certainly in our course over the next few days, about how you might design an antisense oligonucleotide. But locked nucleic acids are one way to do it. It's a very stable, very non-degradable form of RNA that can target other RNAs. So you design an antisense oligonucleotide that is exactly complementary to miR-155. In this particular case, they decided to treat patients with cutaneous T-cell lymphoma to begin with. And the reason for that, and you can see here, is that they were going to first inject the antisense right into the lesions on the patient. So they get these lymphomas growing just under the skin. And so this was a way to sort of ensure that they were going to get the oligo to the right place. And then ultimately they were going to be doing the trial with a subcutaneous injection, just putting it under the skin and letting it disseminate throughout the body. I'm going to show you in a minute that this trial went all the way through phase 1 without any adverse effects found in these patients, and they saw some efficacy. But I won't be talking about this today because what I want to tell you is that prompted the company to move into diffuse large B-cell lymphoma, which is what we were hoping they would do in the beginning, to try to treat those patients. And as you'll see in a minute, these patients have a very poor prognosis. There's almost no good therapy for diffuse large B-cell lymphoma. So I won't be going into all the detail today, except to tell you that we published this work with the company. So that's the company representative there, Amy Jackson. She's the chief scientific officer. In clinical cancer research, the drug was ultimately named cobamycin, but it's that MRG-106, antisense to 155. And in this particular paper, it basically describes a co-clinical trial. So we did studies in mouse, in our mouse models of diffuse large B-cell lymphoma, and they did the study in human patients. Oh, sorry. So basically, I can tell you that we were looking at this most aggressive type of diffuse large B-cell lymphoma, which is called the activated B-cell class. Okay, there's also another class called germinal center, has a different set of genes that are expressed. But we're looking at this really aggressive type, which has poor prognosis. So this cartoon over here just shows you that we and others had already shown that miR-155 was highly expressed in diffuse large B-cell lymphoma. It's also expressed in other types of cancer, but this is one of the highest expressing types. The hope here was that we would be able to treat patients that we knew had high levels of miR-155 in other cancer types as well. Although, as you'll see here, the trial basically started in cutaneous T-cell lymphoma, moved to a few maybe leukemias and diffuse large B-cell lymphomas, but we never really got around to being able to treat these other patients, and I'll tell you why that is in a minute. Right, so the route of injection of the cobamycin drug was essentially either into the tumor itself, if it was a subcutaneous tumor, or it was given subcutaneously for CTCL or intravenously for diffuse large B-cell lymphoma. So the company was also experimenting with different routes of administration. The dose was extremely high. If you've ever done oligonucleotide type work, you'll know that anything in the 100 to 1,000 milligrams of a drug is quite a lot, and what that can do is it can lead to side effects that are off-target effects, not even off-target, just unwanted side effects. But luckily, in this particular study, they showed no inflammatory or metabolic or hematological toxicities, even with these high doses. So this is a pretty inert drug. It didn't do anything to other tissues as far as they could tell. All right, so what this graph shows you is that the cobamycin went into phase 1, 41 patients were treated, and then it went into phase 2, and 10 patients were treated. I'm going to tell you today about these 9 patients that were treated for diffuse large B-cell lymphoma. And you might be wondering why so few patients. I can tell you that the trial never really finished. There was a financial reason for that, and that is that this company, Miragen, was bought by another company, and that company decided not to continue with the trial. But of these 9 patients, 3 of them had the ABC type, which was the one that we were most interested in because that's the one that has the highest levels of miR-155. Our mouse has that phenotype. All of the human cell lines that we had treated, which I didn't talk about, but that are in that paper that I mentioned, the clinical cancer research paper, those are all ABC class of diffuse large B-cell lymphoma. But interestingly for us, one of these patients had a complete response. The lymphoma completely regressed. And why that's interesting is because this patient came on the trial after failing 6 different therapies. So this patient was desperate for some kind of relief, had failed R-CHOP, which is the current standard of care, and failed a number of different sort of experimental chemotherapies, including ibrutinib, which is a BTK inhibitor, which is currently also being touted as a standard of care now. So the physician that was treating this patient took measurements of the 2 lymphomas that the patient had. She had a right cervical lymphoma and a right inguinal lymphoma. And you can see that within just 2 doses of anti-miR-155, those tumors completely regressed. There was no sign of the cancer in that particular patient. But you can also see from this slide that the response was not durable. Unfortunately, those tumors began to regrow within just a few weeks of getting treatment. And in fact, a 3rd tumor, a left inguinal lymph nodes, started to grow in this patient. And so the patient was taken off the trial because obviously they were failing the therapy. Unfortunately, this patient died soon after this particular time. So the take-home message from this is that this drug shows some promise in the clinic, but surely we need further refinements. We don't really understand why this patient's tumor started to regrow. We don't know what the mechanisms of resistance are to oligonucleotide therapeutics. There's very little studies on this because there's so few clinical patients that have actually taken these kinds of drugs. But you could imagine, if I can go back one, that this patient had massive tumor heterogeneity by the time they came on this trial. Probably picking up resistance mutations, deletions, other things that we don't even know because we didn't do a genome sequence on this patient, that could have rendered this patient resistant or sort of finally resistant to an oligonucleotide therapy. So my guess is that tumor heterogeneity was the cause of this. Most of the tumor died, but there was a small subpopulation of cells still present in the tumor that were able to grow out, even in the presence of the drug. So we'd love to understand what these mechanisms of therapy resistance are, but it's unlikely to be some of the obvious things because we don't yet know of any. So you can imagine in small molecule cases, patients sometimes upregulate a pump that pumps the drug out of the cells. We know of no such pump that pumps oligonucleotide drugs out, but that would be one way to think about it. We can imagine getting mutations within a target gene that no longer requires miR-155. So, but we just don't understand what it is. But we do know a few things. One is that we would love to be able to have a more effective drug, and we'd love to be able to target it more effectively just to the diseased lymphoma. So we've been working with an oligonucleotide chemist at the University of Connecticut in the United States, Raman Bahal, and we've tried a number of different technologies now to try and make this drug more effective. And in mouse models and other things, we get much better efficacy, we get much better targeting, but we still haven't been able to take this into a clinical trial yet. It's something that we are trying to get the MiroGen company to do, and if they won't do it, we'll start our own company to try to do this particular work. But you can see we've used some next generation types of chemistries to make these more effective. All right, so I'm going to give you another example of how we're taking sort of basic science discoveries and hope to be able to generate new insights into a cancer type or potentially a new target for therapy in a cancer type. And in this case, we're going to look at EGFR TKI tolerant persister cells. So some of you will know this, especially if you have oncology colleagues, that if you treat an EGFR mutant lung cancer patient with a tyrosine kinase inhibitor, you can shrink the tumor almost down to like 90%, but there's still 10% of those cells that persist. And those persister cells ultimately will grow out, and the patient will either get a TKI mutant, a TKI resistant tumor growing out within about a year of the initial treatment. So we've been trying to understand what it is about these tolerant cells that allows them to basically persist. What causes the tolerance? And is there an RNA that could potentially, tell us something about this tolerant state and maybe even be a new target for that to try and kill those tolerant cells? And work from our lab and other labs has shown that it's metabolic reprogramming of tumor cells that causes them to be tolerant. And I'm going to show you in a minute that we think that we've identified one mechanism for how that metabolic reprogramming happens within these tolerant cells. So what we were able to do is basically we took cell lines. These are human lung cancer lines with EGFR mutations in them, and we grew them on different sort of EGFR TKIs, in particular osimertinib, which is now standard of care, first-line therapy for EGFR mutant lung cancer. And we basically grew these cells in sort of increasing doses of the drug and identified a tolerant population of the cells that we could then keep growing in the lab for forever. And when we profiled these either tolerant or the parental cells that were not tolerant, we could identify which microRNAs were sort of most expressed and which were most differentially expressed between the tolerant and the parental cells. And one microRNA that sort of stood out is miR-21. It's not only highly expressed in all these cells, but it's also one of the most altered in expression between the tolerant versus the parental. So it's upregulated in the tolerant cells. And so we wondered, was miR-21 actually required for this tolerant phenotype? And so what we can do is we can grow lung organoids. We can take these cells and grow them in a 3D culture. And then we can treat them with osimertinib. So this is the old name for osimertinib. This is when it was an experimental drug. So these experiments started before the new name. What we could find is that essentially the lung cells were sensitive to osimertinib, but they could become even more sensitive if we inhibited miR-21 with an oligonucleotide, like we showed you earlier. We could inhibit miR-155. In fact, we could make the cells about 60-fold more sensitive to osimertinib than the parental cells. Now, to show that miR-21 was not only necessary but also sufficient for this phenotype, we overexpress miR-21 in these cells. So you give them more miR-21, and now we could make them even more resistant to the drug. So they're about 14-fold more tolerant of the drug than they were before. So that told us that miR-21 seemed to be critical for this phenotype. And it was our hope that we could now take this information and, if we did nothing else, maybe move a miR-21 inhibitor into the clinic somehow to kill these tolerant cells. But we also went the next step and asked, well, is miR-21 part of the reason why these cells become tolerant? Does it change their metabolism? And in fact, when we did a metabolomics analysis on these cells, these are the cells that are treated with I think this is if can I read it properly, is that the control versus the anti-miR-21? You can see that they cluster in different parts of a PCA plot. And then we could look at the different metabolites that are now found within the cells treated with the anti-miR-21 versus treated with the control. And, without being able to read this, I don't expect you to, I want to point out this metabolite up here called acatazine, which was the most altered metabolite in cells that are persistent versus cells that are now not persistent anymore. So acatazine, if you just look at the sort of pathway analysis of those metabolites that change, the number one thing that changes is purine metabolism. And I'll show you in a minute that acatazine is one of the critical intermediate metabolites in purine metabolism. Okay, so this is acatazine over here. It's also called ICAR. Some of you will know, if you're a cyclist and you follow the Tour de France, that ICAR is a banned drug. Recently, like a few cyclists were disqualified for having ICAR in their blood, because what ICAR does is it activates this pathway and actually activates AMP kinase, gives you more ATP in your cells, but it's also extremely dangerous. You get your muscles become hyper and things like that. So it's dangerous. It should not be taken by anybody. In particular, should not be taken by cyclists wanting to win the Tour de France. But anyway, so what ICAR is it's basically one of the rate-limiting metabolites for the production of AMP and GMP. And what we discovered was that miR-21 actually targets the enzyme that makes ICAR. So ADSL is the rate-limiting enzyme in the production of this pathway, and it's targeted by miR-21 through a sequence in its 3' untranslated region. Turns out that ICAR functions not only at this point in the pathway, but it also, not ICAR, ADSL functions here, but also here. So it has multiple roles in the pathway, and we believe that what these persistent cells are doing is they're upregulating miR-21 to inhibit this enzyme and change the purine metabolism within the cells and actually make them now less sensitive to EGFR TKIs. And so we tested that idea by just asking, well, can we give ICAR to persister cells and change their phenotype? And indeed we can. So if we add increasing concentrations of ICAR, now we can cause these cells to die. So they are very sensitive to the levels of ICAR. And indeed, we could actually sensitize the cells to osimertinib. So there's a synergy between ICAR and osimertinib in these cells, and you can find it over here at this particular concentration of each. So, what we think we've found in this study is that is we found a new non-coding RNA that might explain some of the phenotypes of these persistent cells. We also think that that we've described how this microRNA might alter the metabolism of those cells, and we think then we can maybe take this information to either design a new drug or use an already approved molecule, approved but not approved for cycling, that could potentially inhibit these persister cells and maybe used as a pre-treatment before a patient gets osimertinib to try and kill those persister cells before they even get the drug. So yeah, so we're hoping that we may be able to develop these as potential therapeutics for persisters. All right, so that gives you maybe 2 examples, and we have many more from our lab of targeting a particular microRNA as a new potential therapy. I'm not going to turn for the remainder of my talk, another sort of 10, 15 minutes, just talking about the other side of the coin, which is the potential of using microRNAs as targeted therapies in cancer. So I'm going to talk to you a little bit about microRNAs as potential novel therapeutics. And the example I'm going to give is the potential to use these as immune checkpoint inhibitors. So many years ago now, late 2000s, my lab and other labs found that there was one particular microRNA called miR34 that was extremely, sensitive or responsive to mutation in P53. So, so in many human patients, they get, they delete P53 or they have a loss of function or even some dominant negative mutations within the P53 gene. And that leads to changes in a whole host of DNA damage response genes downstream of P53. One of the genes that changes quite dramatically in the presence or absence of P53 is this microRNA miR34A. And we and others had been studying miR34A for many years before that because it was one of the most conserved microRNAs from C. elegans all the way up to humans. And we knew that it was responsive to the DNA damage. And we'd started to identify targets of this particular microRNA. And hopefully what you can see here is that miR34 directly targets probably all of your favorite oncogenes that you might be interested in working on. It basically has a target in almost all of the Weinberg hallmarks of cancer. And then most recently, we and others have shown that miR34 also targets this immune checkpoint gene called PDL1. And that's involved in immune escape. If you add up all of these things, and I'm not even showing you all of them, there's 40 different known oncogenes that miR34 targets. And we felt like this meant that this must be one of the most important tumor suppressor microRNAs. We also felt like maybe we could deliver miR34 as a way to bypass the loss of P53. So if a tumor has a P53 mutation, we give them miR34, maybe we can restore many of the functions of P53. Our first hint that could indeed be the case comes from this study. We built a mouse that has the KRAS mutation coupled with a dominant negative P53. And you can see that in this particular mouse, these animals, this is a lung from those mice, these animals get lots of tumors and they get large tumors. But if we deliver miR34 to this mouse model, we get no tumors. So this told us that miR34 was probably one of the most important downstream targets of P53, not just a downstream target. You could completely bypass the loss of P53 by giving them miR34. Now, once again, mouse model alert, mouse model alert, mouse model alert. It's not going to tell you what happens in a human, but at least it did prompt us to work with a company called Mirna Therapeutics to design a miR34 mimic as a potential cancer therapy. And so this is the clinicaltrials.gov sheet about this. This is a clinical trial, multi-center phase 1 study of MRX34. This was the drug. It was a miR34 mimic that was basically encapsulated in a liposome to enhance its delivery and enhance its stability. Now, you'll see in red here that unfortunately this study was terminated, and it was terminated because 5 patients in the study actually did show toxicity. We now think we understand why these patients showed this toxicity, and that is because we used a variety of RNA that was not modified enough to prevent activation of the immune system. So we were delivering a double-stranded RNA that was activating toll-like receptors and all your favorite RNA recognition molecules, RIG-I, MDA5, and turning on a cytokine response in those patients. And so what I'm going to show you in the next few slides is what we think we might do if we were to start this trial over again. The first thing we're going to do is we're going to alter the chemistry of the double-stranded RNA. There's no way we would use a weakly modified double-stranded RNA. This has to be completely modified so it does not activate toll-like receptors. And secondly, we would probably do this in combination with some current therapy. And for lung cancer, now a lot of patients are getting an immune checkpoint inhibitor. And I think I showed you in the last slide that miR-34 actually targets one of the most famous immune checkpoints called PD-L1. So our hope is that if we did this trial again, we would do it with modified RNA, and we would do it in combination with an immune checkpoint inhibitor. So what are immune checkpoint inhibitors? So one of the features of cancer cells is that they express on their surface a marker that's not normally found in a cancer cell. This is a protein called PD-L1 that binds to a receptor on T cells. And when it binds to those T cells, it inhibits the activity of the T cells. It basically tells them to back away and not to kill the cancer cell. And so current immune checkpoint inhibitors are antibodies that bind either to PD-1 or PD-L1 and inhibit the activity or the binding of these two proteins with each other. And that now allows T cells to kill the cancer cells. But many of you will know, maybe you've had people in your family that have unfortunately succumbed to cancer and tried these particular drugs, that they are not effective in all patients. They're most effective in melanoma, but you can see that 2/3 of melanoma patients are not responding to single-agent immunotherapy. Now some of them respond better to double-agent immunotherapy. In lung cancer, only about 20% of patients respond, and the list gets worse and worse as you look at diseases like pancreatic cancer, where very few patients are responding. So our hope here was that maybe we could take the non-coding RNA space and try to understand this type of mechanism a little bit better. And secondly, can we identify non-coding RNAs that might sensitize cells to immune checkpoints, to immune checkpoint inhibitors? So I told you that miR-34 directly targets the PD-L1 3' ETR. This shows how a potential duplex might look between miR-34 on the top and PD-L1 on the bottom. There's the seed sequence. You can see that miR-34 targets PD-L1. If you take miR-34 and deliver it into, this is not lymphoma cells, but it doesn't really matter which type of cells you're looking at. But if you deliver miR-34 to these cells and you measure the levels of PD-L1, we can see a dramatic decrease. And that's true at the protein level or at the RNA level. And we also did FACS, and you can show that at the cell surface that it's dramatically decreased. So I'm not going to go into too much detail on that because we did publish that initial work in leukemia a few years ago. What I'm going to talk more about is just a few slides on how we think we might be able to use this if we started a clinical trial, again, a new clinical trial with miR-34. So what this shows you is a co-culture where we've mixed cancer cells with activated T cells. The cancer cells are GFP labeled over here. The T cells we can mark in pink here. This is a CD4 or CD8, CD4 marker over here. If you just mix, so what you're essentially looking at here is the co-culture. You can see the cancer cells mixed in with activated T cells. And over here is a stain for caspase, which is basically cell killing by the T cells. We don't see very much cell killing, and in fact, the T cells are sort of excluded, if you will, mostly from the tumor. But if you treat the cells with miR-34, now what you see is these activated T cells get into the tumor, and actually we see a lot more killing. And so we can quantify that over here, and basically we can show that we can dramatically increase the levels of T cell killing if you pre-treat the cancer cells with a miR-34 mimic, and that allows PD-L1 levels to drop and allows the T cells to kill those cells. So not only that, but we could actually show that we could enhance the activity of an anti-PD-L1 antibody using this approach. So if you give the animals, these cells, two potential immune checkpoints, one an internal one, a cytosolic one, which is the miR-34, plus an external one, which is the anti-PD-L1. Now we see even enhanced killing of these cells. In fact, that's shown over here. So our hope is that we could take this idea maybe in the next iteration of our work and use miR-34 as a way to enhance current standard of care immune checkpoint inhibitors. And we've also taken this sort of to the next step where we're constantly identifying new things that might regulate immune checkpoints. We identified another microRNA that regulates PD-L1. We found microRNAs that regulate ICOS ligand. I mean, if you come to me with your favorite immune checkpoint, we can tell you the microRNAs that probably regulate that, and we could probably design experiments to test whether they could also affect the expression of that immune checkpoint gene. All right, so yeah, this is our most recent work showing that this microRNA that regulates ICOS ligand and can also, when delivered to cells, can sensitize cells to an ICOS ligand inhibitor. All right, so I'm just going to conclude by saying that we've developed nanoparticle methods for targeting oncogenic microRNAs, nano delivery mechanisms as well, where we can add targeting agents to these molecules, and we've delivered things like miR34, for example, in that mouse model I showed you. I didn't talk to you at all about the LET7 work today, but similar experiments with LET7. In the mice, we get no obvious toxicity, but we think that we are going to have to use next generation technologies now to make these more effective and less immunogenic in human patients, which seem to be more sensitive to these kinds of things than the mice are. Yeah, so we still need to understand what some of these mechanisms are, and can we generally apply this in human cancer? I showed you two examples today where we took our basic science ideas and we moved them into the clinic, but not particularly successfully yet. We're still at the early stages. I like to think of where we are now as where the human monoclonal antibody type therapeutics were in the late 1990s, early 2000s, where there was still a lot of immunogenicity toxicities that were associated with that, and it was really a process of humanization and optimization that allowed those to become so successful. I think if we can get past that humanization and optimization step, we're opening up a whole world of new potential therapies that could be useful in a number of different diseases, including difficult to treat cancers like the ones that you guys will work on. All right, so I'm just going to close by just mentioning that we now have this thing called the RNA Collaborative, and I hope that at some point there will be an RNA Collaborative starting here in Sao Paulo. If you want more details about how to join the RNA Collaborative, please let me know. But you can see that there are RNA centers from around the world that now participate, and it's about data sharing. It's about joint seminars. It's just about sharing RNA experiences. So RNA therapeutics experiences around the world. So please start one here and let me know if you want more details. And then I'm going to end by thanking the people in my lab who did the work. These are my current postdocs and students. I've had some great technicians. I've had a number of really great students and postdocs along the way, and really have to thank my collaborators for helping us not only identify new areas that we might want to move into, but also new ways that we can take our discoveries and make them potentially actionable. And with that, I'm going to stop and take any questions if you have. Thanks a lot. You can shout. You can shout.
So very nice talk, but I have two questions. One of the one you take on the lymphoma clinical trial, and the patient respond to your 155 antisense, they found very good. And do you ever check their specimen before treatment to compare with the current? Their MER 155 still maintaining reasonable level, or
right,
it's a different population of the cell?
Okay. Yeah. So now the mic's working. The question was, in that patient that responded to anti-miR-155 treatment and then rebounded, did we ever look at the specimens before and after treatment? Unfortunately, those specimens were not kept, so we don't have the specimens to look at. But if we were to do this trial again, we would take biopsies as often as possible, and we would be. I mean, the original. It was the original. What can I say? The original hope for that trial was that we would do biopsies along the way. but it turns out that that was not done. I think mainly just because patients don't want to give biopsies.
That reality not easy.
Yeah,
Together,
and
yeah.
and but I think it's very important these days in clinical work that you do companion diagnostics, and we think MIRA 155 is an important companion diagnostic for that particular trial. You want to measure the levels of 155. Is it responding to the drug, and then is MIRA 155 coming back on again? So we don't even have blood from that patient, unfortunately, to look.
Second question is also very interesting. You in talk about when you overexpress 34 and stimulated T cell killing. So what kind of 34 you express? Is a mature one or premature one?
Okay, yeah. So the question is when we deliver MIR34 to inhibit PD-L1 expression, what are we delivering? It turns out we're delivering the mature MIR34 as a double-stranded 22 nucleotide strand, two 22 nucleotide strands hybridized together as a mimic. It looks a lot like an siRNA.
Wow.
If you would order an siRNA from Dharmacon or IDT, that we would order something like like a MIR34 from those companies.
Maybe I misunderstand why you say it's a double. You antisense is a single strand, right?
But we deliver the double strand.
You deliver double strand.
Yeah, we
Is
deliver
it possible go through the mechanism because right now we're doing is like a stimulate the double strand RNA. You will stimulate your antivirus pathway.
Right.
So that's maybe the way I saw you dramatically increase your T cell infiltration. Maybe
no, it could be that there's a non-sequence specific sort of adjuvant effect of the double strand. But yeah, we need to deliver the double strand because in order to get into risk, to get into the RNA-induced silencing complex, it needs to be double-stranded. The single strand doesn't get in very well. So we deliver it like an siRNA.
Okay, thank you.
But just so you know, we can modify these RNAs now so that they don't activate the toll-like receptors anymore. So yeah.
On one of your last slides, you showed us how T cells would kill your tumor after treatment with this MARS. How was this treatment actually done? Did you treat the cells and afterwards did you treat it with the T cells, or did you treat the T cells in the tumor with the MIRS? Because these PD-1, PD-L1 axis is not exactly simple, since PD-L1 can be either in the T cell or in the tumor. PD-1 can be either in the T cell or the tumor, and both can be on the same cell, and they can signal differently according to where they are.
Yeah, no, that's a good question. So that turns out that was actually a co-culture experiment using cells, not tumors. So what we did is, you can do this on a slide, but we did it in sort of an like a matrix. You can treat the cancer cells with the MIR34. We wait a day, and then we add them to the T cells. So this was not done in situ in a tumor model, but we would love to be able to try that type of experiment as well. So because we were this was early days of doing co-cultures, we did it in a matrix, but now we just do it on a slide. You can mix the cells together, and it's possible to see the killing that way.
Thank you.
Hi. I'm interested in these persister cells of thymidine kinase treatments that you show that the microRNA 21 is increased. Do you know how that happens? Why microRNA, and whether this can be reversed if you remove the drug, if the persistent cells reacquire the original phenotype if the drug is removed?
Yeah, so that's a good question. So what happens to MIR21 if you remove the drug? I'm sure we must have done something like that, but I cannot recall now what happens in that situation. But MIR21 is there's a fair amount of experimentation about what activates its expression. And it's activated by NF-kappa B. It's activated by E2F. So it's in the pathway of I mean, it's in the KRAS pathway. But yeah, what happens if you remove the drug? I don't recall that right now. Sorry.
Hi, very interesting talk. First of all, I was wondering about the resistance mechanism. Could it be that maybe cells might upregulate RNA modifying enzymes to modify the microRNA or their targets? Because I imagine if you're delivering microRNAs to cells, it might be overwhelming the RNAi pathway and maybe interfering with other microRNAs and their targets. So maybe the cells could respond by upregulating enzymes that would modify the microRNA and preventing it from being processed or binding risk or even recognizing their targets. Or alternatively, the targets might be modified.
Yeah.
So this could be a potential resistance mechanism.
Yeah, for sure. Yeah, so, we have a list of the things that we would probably want to test. At the moment, we don't have any experiments to really look at that. But yeah, I mean, you probably know that most tumors have downregulated levels of microRNAs because they have very, very reduced activity of DICER and DROCHER and some of those enzymes that process microRNAs. In fact, there was a paper just recently showing that the KRAS pathway alters methylation of Argonauts and DICER and things like that changes their activity. So it could be that in these patients they activate. It doesn't have to be even RNA modifying enzymes. They could be activating protein modifying enzymes that alter the levels of these processing enzymes. But yeah, RNA modifications is something that we've only just begun to start looking at in my lab. We've had maybe 3 or 4 papers looking at sort of RNA modifications. I would say MIR21 has never been one that's come out of any of the studies that we looked at to be modified. But LET7 does get modified in a number of different ways. So certain microRNAs are modified that way. In terms of the RNA of the microRNA binding sites, those can get modified by RNA modifying enzymes as well. And so I think when I said earlier that one mechanism of the resistance here might be that the tumors either delete the microRNA binding sites in key downstream targets or they mutate them. It could equally be possible that they're just modifying them so they can no longer respond to the RNA to the microRNA. But we don't know that yet. That's a good point.
Just another quick one. So you mentioned that the MIR34 was basically a double-stranded small RNA, like an siRNA. So why not use a PD-L1 siRNA as it would be, I assume, most effective in silencing PD-L1?
Yeah. So the question was, if you wanted to target PD-L1, why not just target PD-L1 with a siRNA as opposed to using something like a microRNA? Well, I think that's an excellent point. And in fact, there are experiments out there, and there are companies out there trying to develop those kinds of drugs. The way that we see MIR34 as being potentially beneficial is that you're not only affecting the levels of PD-L1, but you're also affecting 40 different oncogenes, including MYC, BCL2, CDKs. I mean, the list goes on. So we think that it may have a benefit as sort of a one-drug cocktail. One drug is affecting 40 different things as opposed to just one thing. So yeah, the danger with these kinds of experiments, of course, is that you could also have unwanted downstream targets that maybe the cancer cell actually doesn't want to target all 40 of those things, but only 20 of them is good and the other 20 shouldn't be touched. But these days combination therapies are so common. I mean, you get patients that are getting 5 or 6 drugs at once. Here we have one drug that could be affecting multiple things at once. So it's a dream, and it may not be the right way to go, but we think that it's potentially beneficial to target many things as opposed to just one.
Very nice talk. Congratulations. I was curious about your the first experiment that you showed, which was really impressive to see how you got this widespread lymphoma with the overexpression of the MIR155. Did you the way you did it, did you activate the expression specifically in the lymphocytes, in hematopoietic cells, or in the whole body, the whole cells, I mean, the genome? Because you did a transgenic, to probably use the Cree recombinase or something.
Yeah, no, that's a good question. Sorry, I forgot to mention that. This was activated in the hematopoietic system. Yeah, and
the other thing was about the clonogenicity. I mean, I was wondering whether you had multiple events of lymphoma formation in each lymphoma, like each tumor would be one clone, or there was basically just like in the disease one.
Yeah, that's a good question. So what about the clonage? Were the multiple clones that came out of in the same mouse? We actually usually only found one tumor per mouse, and then if we let them go a little bit longer, we could show that metastasized. And we actually do understand what's happening there. So we identified from those tumors that miR-155 is regulating a number of different factors. It actually regulates genes for DNA damage repair. So when miR-155 gets activated in just one lymphocyte, it cannot repair DNA damage. The second thing is it actually also represses a number of tumor suppressor genes like SP1, PU1, MIB. So there are multiple things that it does to those cells to survive. And when we did a whole genome sequence on those tumors, every mouse had a different set of mutations. So every tumor that came from mouse 1 had a different set of mutations than the tumor that came from mouse 2, except for in one gene. They all had an activating mutation in KIT. So KIT is a tyrosine kinase inhibitor. And so what happens is in these mice, they probably pick up mutations all over the genome, but they can't repair them because they're repressed DNA damage repair genes. And then the one mutation in KIT emerges and the tumors start to grow. And so if we give them erlotinib, which is something that inhibits KIT, the tumors will regress. So yeah, so this was an interesting way to try and we would try to understand why these tumors form and why they're sensitive to miR-155 and why they're clonogenic.
Very interesting. Thanks.
Sure. All right. Thank you very much, everyone.
May I do the last question?
Okay.
Very quick.
Can we talk in the coffee break because we are late, and a short 30 minutes coffee break will be served at Finesse restaurant in front of crossing the street, and please come back in 30 minutes. Thank you. Pessoal, pode filmar a lista de presentes. Aqui não assinou, a lista está aqui.
So you probably know that most of the So that has changed the efficacy and changed the whole landscape of RNA drugs to the point where everything we do now is probably going to have a targeting that you have to get it to the right place. So for something like Mir34, we initially put it into a lipid nanoparticle thinking it was going to be protected and cover everywhere in the body, but mainly goes to the liver.
Oh,
Yeah, so
you inject in IV.
Injection IV, it goes to the liver. So it wasn't very effective. Juicy work we catch proteins and it goes to some protein So that's for an antisense molecule, you target it with peptide or carbohydrate. You've tried lipids that don't work as well. Generally, carbohydrate will get it to this. So lectins that are expressed on particular cell types. If you know what those lectins are, then you know what they're binding to. You can put that carbohydrate onto your RNA and it will get into those cells.
Mm-hmm.
So that's what we do.
Oh,
So,
okay.
but there's very little clinical work now. We would like to start more with that.
So you also work to develop this kind of a system, or you have kind of collaboration?
I have collaborated because I'm not a biomedical engineer.
Because it is
I'm not organic chemist, yeah, so I have collaborated.
I see, yeah.
But if you have things that you want to try yourself, I'm happy to put you in touch with my collaborators.
Thank you very much.
Yeah, sure.
Really nice talk.
Yeah, thanks. You guys want to walk over and get coffee? Or you can...
Yeah, let's go. Ô, Sueli, tudo bem? Quanto tempo? Eu acho que vi você, mas eu estava pensando
Can you
que...
get safe to leave bags? Leave bags?
I'm just waiting my Dr. Singh is my professor.
What are you working with? Okay. Okay.
Pode deixar. Me deu um branco, acho que é isso.
Esse aí, esse aí. Uhum. Hi, let's start. I would like to introduce Dr. Gangning Liang. Dr. Gangning Liang is a professor of the Department of Urology and Director of Genetic Translational Research at USC Institute of Urology at Norris Comprehensive Cancer Center in Los Angeles, California. He obtained his MD degree from Shanghai Medical School and received the PhD degree from the Department of Pharmaceutical Science in the University of Nebraska Medical Center. He completed a postdoctoral training in epigenetics with Dr. Peter Jones at University of Southern California. He has enormously contributed to understand the roles of DNA methyltransferases during tumorigenesis and novel methods of characterizing epigenetic changes and monitoring these changes in tumor patients. His work is focused on translational research to use epigenetic alteration as foundation to identify drug targets in a selective and predictable way for precision medicine and to improve patient survival and quality of life. I have the pleasure to invite to the stage Dr. Gangning Liang. Thank you.
Thank you, Maria, for a nice introduction. And indeed, I already here for 10 days. I really enjoy Sao Paulo, the city, the environment, museum, park, of course, include the food. Okay, yeah. Today, my talk majorly is say the SETD2 deficiency and how we take advantage of the SETD2 deficiency as a therapeutic target for, especially for the aggressive kidney cancer. So this is my disclosure. So I'm not original think about study SETD2. And we just based on our previous early study and somehow based on our hypothesis leading me to study the SETD2. So this is the study and published in 2012 and by Steve Bailing's group and in Johns Hopkins. What did they do? They use the DNA methylation inhibit. You know, we are epigenetic, usually regulated by DNA methylation, histone modification, and the nucleosome occupancy and the non-coding ion. And in this is the DNA methylation. And especially the drug have very short half-life, like 30 to 60 minutes. And what did they do the experiment in this one? They treated the cancer cell for 3 days in very low dose. And after that removed, and they continue culture for 1 or 2 weeks. Then they injected to the mouse. Compared with the untreated cell, the tumor volume is very dramatically decreased. So this period around 2 months. Then they could take that mouse, inject to the next mouse. and the tumor continue to be inhibited. So in general, 3 to 4 months, and the tumor still show the anti-tumor memory. That is their key information, deliver the novelty discovery about anti-tumor memory by this DNA methylation inhibitor. However, this drug, they see several months ago, only have less than hour half-life. So in that paper, they published detail about the transcription and which potential pathway to target. However, they didn't really digging in how the DNA methylation happened after treated with the DNA methylation inhibitor. So I take advantage the one they didn't do this portion. So I did a very simple experiment trying to mimic in the cell culture. So what I do in the several cell line, and we treat the drug for 24 hours, low dose, and remove. Then I just watch the cell doubling time. You can see in the first period of time, they have dramatically inhibited. They gradually go back to the untreated cell. So that's also similar, take 2 months. Somehow phenotypically, we confirm their discovery in the mouse model. So then in detail, I digging out the DNA methylation. I think if somebody take the class last week, I said, in the field, people always think you treat with the DNA methylation inhibitor, you remove, they quickly rebound. That's it. So most people think about nothing we should study, but keep doing it, and we found it very interesting. So we found that majorly have 2 groups of this is a DNA methylation array. Okay, the yellow means they heavily methylate. We asked the heavily methylated locus and low sites, and after treatment 24 hours, day 5, day 14, day 24, day 42, how did methylation behave? The rebound. Remember, we only treated 24 hours. We only treated 24 hours. We don't really have anything else. So you can see day 5, you show the strong demethylation, and then gradually methylation go back. And that's it. And the majority of the group actually never really go back. And that time we are really interested in which region is rebound. We found that it's very interesting, and also it's undiscovered before my study. This rebound region actually on the transcriber region. If you study the DNA methylation, you know the first concept you have a DNA methylation, you will think the silent DNA methylation promoter and the gene silent, you treat the drug and they reactivate. And we found this one highly enriching transcriber the region. Second, this group of gene always overexpress. So that's we found. Then next we asked, and which methyltransferase involved? Methyltransferase have a DMT1, 3A, 3B. So just to cut the story short, we actually found that when we knock out the 3B, there's a fast rebound group, and the rebound much slow. Then we put a different 3B isoform, and you see somehow restore the DNA methylation, but not in this group. You see, you manipulate all the different type of the methyltransferase, doesn't really change the methylation pattern. So next question we ask, you remove the DNA methylation, what makes 3B go back to the same place, right? So we actually, and this is the methylate and methylate. We ask the question, when the methylate or unmethylate, which the different? We found a K36 trimethylation. It's not dependent on your methylation status. They're always there. Also, they're enriching the transcriber region. So that's when we make a link. Quickest restore the DNA methylation by 3B, 36, and then restore the methylation. So, as I mentioned before, and in general, and the gene, normal gene is expressed, and the promoter region is not methylated. And the transcriber region, you can see some residual methylation. And however, the promoter region methylated gene is silent. And in our this study, we found overexpressed the gene, the transcriber region actually heavily methylated. So now if you treat it with the DNA methylation inhibitor, traditionally people think remove this one, such as a tumor suppressed gene can be reactivated. And by our study indicate a lot of oncogene, like a MYC pathway gene overexpressed, the transcriber gene region was heavily methylated. If you treat, the overexpressed gene could be downregulated. So that is the first time we found the DNA methylation actually have a dual role. Not only can reactivate silent tumor suppressor gene, also can downregulate the potentially overexpressed oncogene. And we also found that this link, and because of the 3B K36 SETD2. In addition, and I also will discuss later, so with our collaborator group, in 2015, we also found it's another third novelty of the DNA methylation inhibitor. They can turn on endogenous transposon element. In general, they're silent by DNA methylation. And most of them should think about is non-coding RNA. And some of them produce the double-stranded RNA stimulate immune response of our cell. In other words, just make our cell express virus. Make a tumor cell, especially tumor cell, catch cold, then you stimulate our immune response, increase the T cell infiltration to the area. So that's just the detail. I think, majorly, because right now this concept is pretty hot, and endogenous, the region silent by DNA methylation, the transposon elements are silent by DNA methylation. When you treat with the DNA methylation inhibitor, you express a double-strand RNA, and then stimulate antivirus pathway and increase the conversation between the T cell, increase the T cell infiltration. This is just another figure how to explain, and they can increase the T cell infiltration. Then you can then plus the immunocheckpoint inhibitor Frank just mentioned, and you can synergistic effect for immunocheckpoint inhibitor treatment. So now, as I mentioned, and the original based on our publication, we think, and when the region you transcribe, you methylate, and you treat it with the DNA methylation inhibitor, and you remove the methylation and the transposon element, because the transcriber region contains a lot of transposon element. We think the transposon element can be expressed and produce a double-stranded RNA, stimulate immune response we call viral mimic. However, and this system, because you have a K36 trimethylation, which is the SETD2 is a methyltransferase of a K36, and 3B recognized K36, they can restore the DNA methylation. Then the transposon element will be resilient. Okay, so that's we think now based on this one. Then I'm thinking about if any type of the cancer and SETD2 deficient, and you will expect same thing. You treat, you activate transposon element. However, because you don't have a K36, and you maybe cannot restore the DNA methylation, so then you have accumulation of the transposon element, and that will be and further stimulate and the viral mimic. So that's a kind of the idea is maybe SETD2 is the cold tumor. We treat with the DNA methylation inhibitor, induce the transposon element, stimulate the immune response, increase the T cell infiltration, become a hot tumor. However, the hot tumor is the therapeutic target for immunocheckpoint inhibitor. So now the story, I have to looking for SETD2 deficient. So SETD2 have so many potential functions. Besides the key is recognize K36. They also directly involve the antivirus pathway. This is a key STAT1, the activated form requires SETD2 to methylate the protein. In addition, as we said, and the SETD2, because it's played a role, include our own data for the rebound methylation on the transcriber region. That's we think is a potential therapeutic target. And also, and SETD2 involving the DNA methylation, DNA repair deficiency, also the splicing. So I will mention all of this later, how we attack their weakness, become our therapeutic target. So now we look around and so which cancer is the most common? Answer of them is the kidney cancer. 15% of them have a mutation. So that's pushed me go back to my original department. I'm from the urology department. So people always ask me, your urology department, why you do liver, why you do the skin cancer or pediatric glioma, and also do the retinoblastoma. I do all this kind of thing. So that's because of methylation happening in all this kind of disease. That's why I'm gonna go through all type of the cancer. So we focus on this one, K36, and I have the very senior member in my lab, Hongtao Li, and he did a great job. He not only do the wet portion, he also do the computational analysis. So the kidney cancer actually is not really aggressive cancer. Most of them it have very good survival, and in general, it's around 6 to 70% survival. However, when you go to these stages, the recovery, the survival is very bad. So in general, this one is a mutation, is the highest in kidney cancer. Another study show they analyzed the first four most common of the mutation in different kidney database. You can see in general, this four no big different except this one. This database is a metastatic kidney cell. You will see the metastatic or recurrent kidney cell, the SETD2 is a dominant mutation. That's indicate SETD2 may be involving the aggressive of the kidney cancer. Besides the mutation, we also found based on TCGA data set, and the SETD2 in general is downregulated in the cancer. And the downregulation of the SETD2 also in general correlated with the tumor stage. More high stage, the one have a low SETD2. And also is really have a better survival beyond the mutation. If the SETD2 level is low, your survival rate also is bad. And also, so this is another study based on 600 patients. And what they do, they found, and SETD2, they use the immunostaining. They check 600 of the kidney cancer patient, use immunostaining, they found a low SETD2 is really correlated with a better outcome. So another study, also based on the TCGA data set, they found that the patient with a high level transporter element expression actually very sensitive to the immuno checkpoint inhibitor. So that's encouraged me to do the further more detailed study. And what in here I did is the one, this is a high level SETD2 patient, low SETD2 patient expression, also the mutated patient. We separate these two groups, ask them the transporter element expression level. Indeed, supported the previous study. So high SETD2, you have a high transporter element expression. That means they're sensitive to immunotherapy. And the low or mutant ones, relatively low and maybe not sensitive to immuno checkpoint inhibitor. So that's the one. So now the idea coming. So can we treat and turn on the low to the high, then you can, in combination with the immuno checkpoint inhibitor, the treatment. So now in this one, we're trying to attack this one. We can restore antivirus pathway, and we also hopefully block the DNA methylation, can have more transporter element to express. That is our idea. So once again, and I show this one, that is a whole idea. Hopefully mutant will have a more transposon element to express and convert the cold become hot. And the DNA methylation, in addition, DNA methylation inhibitor is already in the clinical trial. They're very efficient for liquid tumor. However, they have some problem right now for the solid tumor. So that's why recently, almost 3 to 5 years, any grant I submit use target the solid tumor, always have a keyword from one of the reviews say, yeah, clinically they never see DNA methylation inhibitor work. So that's just kill my grant. So I'm really disappointed, yeah. However, recently have some new drug developed from GSK. The first they found for the oral drug, and also GSK have a new compound. The half-life can maintain to 24. And leading by Peter Jones, Steve Bailey, the Stanford Cancer team, they begin to show some response from solid tumor. So we go back and we first pick the kidney cell line and the wild type and mutant one. And some of them are heterozygote deletion, some of them homozygote deletion. And you can see the SETD to target the K36. The mutant one, in general, you have a low-level K36. And we also test the DNA methylation inhibitor half-life, and it's very nicely, you will see white mutant one or increase the half-life is short, especially sensitive to DNA methylation inhibitor. And now next we treat this cell line, and this two is a wild type, this two is a mutant cell line, endogenous mutant one. You can see in general, one of them behave really good, and the antivirus pathway gene is strongly upregulated. This one is a heterozygote deletion, but still shows some response compared to this one, not dramatically. But remember, this all have a, besides SETD2, they all have a different genetic background. Other factor called involved. So we knock out the 7A6, and so now this two cell line, the only different is a SETD2, right? So you can strongly upregulate antivirus system after you treat it with DNA methylation inhibitor. Actually can maintain pretty long, day 5, 15, 23. So now next we ask the transposon element, how they express. Clearly we can see in the SETD2 knockout cell, you have a much strong transposon element expression than the wild type, but they on different location. In addition, you remember original theory I developed because I'm thinking the rebound methylation and maybe played a role to resilient the transposon element. When you knock out the SETD2, you cannot rebound, and maybe that's one high. Surprisingly, they both rebound. You knock out the SETD2 even higher. So totally kill my hypothesis. But the phenotype still stand on my way. They're sensitive to the drug. The mechanism go another way. So the mechanism actually, we found something pretty novel, and then I will not present it today, but hopefully after 1 year, I can have some exciting data to report to you. So then we ask the question, why this region is strongly reactive? We ask the K36, not surprised. You knock out, you treat the K36 low, here start with the low. No explanation. K27 trimethylation, no explanation. And the K4 trimethylation, K27 acetylation. Both didn't give us a clear answer. So, but we found a very interesting on the K9 trimethylation is really high in this group. The transposon element actually express. So that is another shocking for us. You know, K9 trimethylation is a repressor mark. People will know this is when it's a repressed transposon element. How do you gain the K9 and your transposon element further express, right? So we spend several weeks to think about, go to the PubMed, do the research, and we finally found a paper show, and actually not only one, several papers indicate when you have a enrich of a K9 trimethylation involving the missplicing. So this region is maybe highly misspliced. So during the time and several, two of the Cell paper actually used the splicing inhibit and to try to get intron inclusion. And because intron enriched with a transposon element, and they use this advantage to stimulate immune response and the combination with a checkpoint inhibitor treatment. And also this is a similar approach, but they're looking for intron using the small peptide. That's also think is a tumor antigen to stimulate the immune response. So by this suggestion, we ask ourselves, is it possible we also have the missplicing? So indeed, we found in the DKO cell, in the knockout ZD2, they have highly enriched splicing in a whole different form. So for example, the typical example is this is the Y type, and this is the transcripts, right? The exon here. You will clearly see you after treating, you increase a little bit. But when you knock out, besides exon, you see the intron region expressed. And the intron region, this is a repeat mask. You can see the repeat sequencing is enriched. So that's the way we think. Now you include the intron, express more transposon element to the system. That's maybe explain why and stimulate immune response. So here is the data in the Y type and the ZD2 knockout. We can see after we treat with AZA, this two of the immune response, the signal is very strong in the ZD2 knockout. So what we're trying to do is we get this isoform overexpressed in the Y type. See, we can stimulate this one. So we pick A of them, at least half of them show the immune response is strongly upregulated by this isoform cell. So now this one is very, now based on this data, we think the mechanism is in the Y type, you treat, you can turn on the transposon element. And however, in the knockout cell, because you enrich of the K9 trimethylation, and not only you express endogenous the transposon element, also you induce the missplicing, and the missplicing carry a lot of the transposon element, double-stranded RNA, and stimulate the immune response. So now somehow we indicate that this potentially you can increase the cold tumor become hot tumor. And the key question is, we ask the question, is that will be help increase the immune checkpoint, the inhibitor, the treatment. So in the mouse model, we have to use the competent mouse, and then we use the mouse kidney cell line. We knock down the ZD2. We can see lost the K36. We also show them sensitive to DNA methylation inhibitor in vitro. And then we did a mouse work by collaborate and see the science group. And in the just cut the story short, and we treat DNA methylation inhibitor alone, and we treat the PD-L1 inhibitor alone, and the combination. So in the combination group, you can clearly see the synergistic effect by DNA methylation inhibitor and the immune checkpoint inhibitor. And we also can see the enriched T cell infiltration in the tumor. So the conclusion is that I think I tried to save some time, and the conclusion is ZD2 is a cold tumor, and use DNA methylation inhibitor potentially you can turn cold tumor become hot tumor by induced missplicing, and then you can get in vivo we confirm potential the synergistic effect with the immune checkpoint inhibitor. And so this one right now we're looking for the clinical trial because the drug efficiency and make the Stanford cancer team a little bit hesitant. They're trying to waiting for, and right now their result, if it's good, they maybe will take this one as a potential clinical trial. So now, as I mentioned, the ZD2 have so many defects. another defect is the DNA repair. So now I'm thinking about, we already confirmed DNA methylation inhibitor can sensitize SETD2. Can we also attack the DNA repair? The mechanism of the one is a K36. When the DNA is damaged, K36 play the role to recruit the DNA repair component to repair the DNA damage. If you lost the K36, you will have a damage, but you fail to repair. So that will be potentially induced apoptosis or when during the treatment. But if it's not a treatment, that's just increase mutation frequency. So indeed, and the people already take advantage on this pathway, and majorly they are focused on the BRCA1 and the BRCA2, and AD did this kind of repair and use the PARP inhibitor, and to increase this as a therapeutic target for PARP inhibitors. So can we replace the SETD2 as a PARP inhibitor target? So PARP inhibitor hasn't been commonly used on all this variety of cancer, but not really in the kidney cancer. So the Steve Bailing's group also published an interesting paper. They show and they use the PARP inhibitor, and you can increase the DNA damage. However, if you combination with the DNA methylation inhibitor, they will trap not only the PARP, also DMT, and get more region be damaged. So that is also show the synergistic effect with the combination treatment DNA methylation and PARP inhibitor, especially for BRCA type of mutation. So now I think I can exactly take the same model to check the SETD2. So now we, as I mentioned, we already proved DNA methylation targeted this pathway, potentially this one, and also the splicing. Now we add a PARP inhibitor, we maybe can target more pathway. So this is Xin Yizhou, is a MD PhD student, and we co-training with one of the university in China. So just as I mentioned before, the sensitivity in the cell to the DNA methylation inhibitor, one of the very important is the cell doubling time, because there's a passive incorporation of the drug into the newly synthesized DNA. So if you compare the cell line with a different doubling time, it's very difficult to figure out the sensitivity of this drug. So in here, what are we just trying to not confusing review too much, and we use the same doubling time, similar doubling time of the wild type cell and the mutant cell. We want to see the difference. From here, we clearly see the mutant cell is sensitive to DNA, not only sensitive to DNA methylation inhibitor, and also more sensitive to the combination treatment. Another comparison, this is the one cell knockout, and also, and you lost the SETD2, and they were not only sensitive to DNA methylation inhibitor, also with the combination with the PARP inhibitor. So we also prove, as say, gamma H2AX is dramatically increased in the SETD2 deficient cell, especially in the combination treatment. And some of them, for example, this cell line Y type, and in the mutant one doesn't really show the dramatic difference, but we also did a COMET assay to check the degradation of the DNA. We can see, doesn't matter which one, we all see the significant increase of the tail in the SETD2 deficient cell by combination treatment. So that is once again indicate combination treatment could have a synergistic effect for SETD2 deficient cell in kidney. So another thing, as I mentioned, and lost the K36, you may lost the recruitment of the most important DNA repaired component of the protein. So we did the Western blot, And from the chromatin, you clearly see in the knockout cell, this kind of recruitment, even previously, maybe this is slide show. Yeah, previously you can see the gamma H2X is high, but the recruitment for this repair component actually is weak compared with the wild type. And so that's indicate, so in the SETG2 mutant cell, you will get more damage, but you have less recruitment for the repair. So the consequence will be induced more apoptosis. So also, and we found that the combination treatment also can turn on the endogenous viral gene. It's not surprised by the DNA methylation inhibitor. Because much more of the apoptosis cell to induce, they also can stimulate another important immunopathway. They can also turn on by endogenous virus. You can express DNA. This is RNA pathway. This is DNA pathway. They also can upregulate the STIM pathway. So here, just the example, you can see in the SETD2 deficient cell, DNA methylation inhibitor treatment alone or combination, the STIM pathway is strongly upregulated compared with the previous status, also in the knockout cell. And also not surprised, immunoresponse pathway is upregulated, and I think majorly because the DNA methylation inhibitor turn on endogenous virus gene, and you can see the strong upregulation in the combination treatment of the endogenous virus gene. That's what contribute to stimulate immune response. And we also found that the downregulated gene is enriching the chromatin assembling gene. So that means when you set G2 deficient and the cell is not chromatin is destabilized and is more vulnerable to the transporter element expression, DNA repair deficient, and induce the apoptosis. And that's just confirmed by the data analysis. See, this is key of the pathway is strongly downregulated in the combination treatment. And this is just several individual example show the H1 is really high in the combination treatment. It's really downregulated in the combination treatment. H1 is played a key in the link, right? And this is Western or indicated. So the chromatin and the chromatin stability and play the role to silence the transporter element, and also play the role for the stabilize the DNA methylation and DNA repair and genome stability. When you lost this one, not surprised. You can see transporter element upregulate, DNA repair efficiency is decreased. And also this is a review paper indicate, and when you induce the cell apoptosis, you also can stimulate immune response by release the debris of the DNA, also transporter element, once again to stimulate immune response and stimulate T cell infiltration. And here is another one based on TCGA data set. We found most kidney cancer and this group of gene be upregulated. After treatment, we can see like 5 or 6% of a gene can be restored by the combination treatment. But the most downregulated gene actually more efficient, more response to this kind of treatment. You can see around 20 to 30% of a gene can be restored by the combination treatment or DNA methylation treatment alone. So finally, we did a mouse work and used the DNA methylation inhibit and PARP inhibit. Both is a chemical drug. Sometimes the toxicity is people concerned. And so in here, we just show the mouse weight and the wild type and tumor, no big difference. In general, we use those is tolerable for in the mouse. And we also confirm our cell and it's really lost the K36. And this is the mouse work. Clearly we can see And this tumor is a response to the DNA methylation inhibit, and also with the combination treatment in the knockout cell. However, one of the questions is a review. Actually, we answer ourselves, but we don't have a clear answer until we send a paper. Review force us to answer this question, and the tumor size actually no significant different. It's a why, right? Based on our in vitro, definitely should be sensitive, and so we did a further study to answer review's question. And first we found the K36. That is a proliferation marker, and so you clearly can see in the combination treatment is down. So that's my cell is not aggressive as the wild type, and not like ASA alone. And the combination treatment, you have a further cell inhibition. And we also checked the apoptosis cell, and we found that in the KO cell have a more apoptosis cell in the combination treatment. So we later get a tumor and the later pathology check. Indeed, we actually found in the combination in the KO cell have a lot of necrosis. That may explain why the tumor size doesn't really change. So that is a conclusion, and regarding this one, and because by combination treatment with the PARP inhibitor, we will accumulate DNA damage insufficient to repair. Also, the upregulated immune response by viral mimic and the STIM pathway. And also lost the genetic instability, also push this pathway, get increase this pathway to unfavor the tumor growth. Okay, as I mentioned, during last 5 years, I really not happy with my grant situation. Each time I apply, they always say DNA methylation doesn't work. But I already spent 5 years on this field. Then I say, oh, can I find some drug? Nothing to do with DNA methylation inhibitor. So I searched, I found that it's called WEED1 inhibit, and the target on SETD2. They also can produce the double-strand RNA, stimulate immune response. Then I say, okay, maybe I can use this drug to replace the DNA methylation inhibitor. That's the way. So we have the visiting professor from Japan, and what he did, and he found the cell line sensitive to WEED1 inhibit. 2 of them is the SETD2 deficient cell, but another one is wild type. And we also found the resistant to the WEED1, and the 2 of them is the mutant one, and 1 of them is a wild type. Okay, 2 of them is a wild type. So from this data, doesn't really tell us the previous publication, something is misleading or something we're missing. So we further do and use our cell line. We knock out the SETD2. We clearly show the sensitivity to DNA methylation inhibit, sensitive to combination treatment. But when you knock out, not like the Cell paper previous report, they're not sensitive to the SETD2 knockout. So something else will be happening, right? So what do we do? And I just say, okay, we do RNA-seq, what we can do? And so just cut the story short. So we actually found that compared with sensitive and non-sensitive, P53, the driving pathway, is make the difference. So that is my future study. So right now, working on. But at the same time, suddenly one of the grant will be found. So I still have a chance to work on DNA methylation inhibitor. But this is my backup plan in case the grant will be rejected again. So that is our research life. Yeah. So that's the future direction, because based on the result, I work in urology department, working cancer center, a lot of clinical doctor interested in this kind of clinical trial. So right now I'm giving them a choice. And the first choice, of course, we're working with Peter Jones and Steve Bailing, the Stanford Cancer team, because they have money ready to fund for any type of the epigenetic drug for clinical trial. So that is my future plan. And hopefully one day some of them can go clinical trial. And also, as I mentioned, is right now another hot part is so intron inclusion. Besides, we're talking about the transporter element, and also people really interested on this kind of short peptide to stimulate. That's maybe one of the future direction I want to working on. Yeah, finally, and that is my lab member during last 10 years I'm working on. And this is the picture we took the before COVID. And this is Deborah and from Marian's lab. And I think she really enjoying the Los Angeles life. And that's all my collaborate and the funding support. And so that's it. Thank you very much. Happy to answer any questions.
We're now open for questions. No question?
I have a question.
Thank you.
General question.
I mean, inducing this viral mimicry may also induce genome instability, as you pointed out. So I guess that could be not that very tumor specific. So what do you think, is there a therapeutic window that would be to maybe narrow to work to explore this therapeutic opportunity of SET2 deficiencies? Or
Yeah, that's a very good question. But at the same time, we remember this not like Dr. Frank just say microRNA more specific or the specific. This is a chemo drug, and the chemo drug is in general as majorly they target fast-growing cell, just like most chemo drug, and you definitely will affect the normal cell. And we lost hair, we vomiting, and chemo because that reason. Yeah, so however, remember the DNA methylation inhibitor. I'm sorry. So remember the DNA methylation inhibitor, and that's Peter Jones' contribution in the early 80s. And the people found that this DNA methylation inhibitor is a chemo drug. It's very high dose, but it doesn't really work until Peter Jones stepped in this field, and he actually did the one low the dose, go extremely low dose, and show its toxicity is not the major driving force. Majorly is a demethylation. So that's in the last 10 years, this drug will be reintroduced into the clinic.
And just another quick question. Sure. If there's any information when you people that have been studying the SET2 deficiencies in terms of tumor heterogeneity, are they? I mean, do they affect every cell in the tumor, or if there any information regarding the?
That's a very good question. So the Frank just previous one just mentioned, in his mouse model and my mouse model looks nice, but when you deal with a real human tumor, the heterogeneity definitely is the problem. And so definitely that's in the clinical trial, and we have other clinical trial. The key is a specimen, before, after, recurrent, non-recurrent, then can give us figure out and what is the real efficient drug target, or what's the recurrent. Could be have a second or third target. Hopefully the combination can kill all of them.
I guess maybe the question is how much this tumor mutations are present, for instance, in tumor stem cells, or how heterogeneous they are in terms of the...
I don't have this kind of detailed information because first, this mutation rate is not really high. Right, 15%, and we haven't really do detailed, for example, single RNA-seq or this kind of analysis. It's very difficult to answer, but if you can imagine, definitely should have a wild different. Yeah.
Thanks for a very interesting lecture. I was wondering if there is a standard way of measuring if you have an upregulation of these transposable elements. What is the standard way to say, oh, I changed the cells in some way and I'm inducing these transposable elements, and then these therefore is important for immunogenicity as well.
Yeah.
Like in vitro.
In vitro.
In cells, yes.
In
cultured cells.
vitro ED, you just before after treatment upregulate, you show phenotype change. You will think that's the direction you turn up.
But how do you measure these transposable elements?
Oh, that's by the RNA-seq, but you need a total RNA-seq. And if you do coding because you do the poly-A, most of them don't have a poly-A. Most information is you missed. But in the clinic, I think last week I presented one of them, show response to the drug treatment, non-response. And we just do the RNA-seq or RT-PCR, compare the response, non-response, before, after. And you can see the response to the patient. You can see strong upregulation of the transposable element. But we haven't seen that one in the non-response patient. So that's actually answered the question. And we with a collaborate in 2015, they actually want to see some evidence from the clinical trial. And we don't have until the paper published after 2 years, we finally get a clinic data on the patient's AML. Remember that drug is very sensitive to liquid tumor. So we finally have a sample to prove that we right now describe the mechanism. It could be happen in the clinic.
Any more questions? No. Thank you, Gangning. Very nice. Thank you. So I would like to briefly introduce Dr. Jeremy Nzengimana. Dr. Jeremy Nzengimana is Senior Lecturer of Biostatistics in the Population Health Sciences Institute, Faculty of Medical Sciences at the University of Newcastle. And he holds a PhD from the Université Catholique de Louvain, Belgium, and has previously worked at the University of Leeds School of Medicine in Leeds and the International Agency for Research on Cancer in Lyon, France. Dr. Jeremy's research focuses on efficient design and analysis of preclinical studies with a particular interest in biomarker and drug target discovery. He is a member of the Testicular Cancer Consortium and the Melanoma Genetics Consortium. Dr. Jeremy, please join me at the stage.
Hello, everybody. It is really a pleasure for me to be here. Thank you, Miriam, for inviting me and giving me this platform. So I am Jeremy, and I'm going to share some of my work in melanoma biomarkers. So I wanted to talk about myself, but Miriam has already, for some reason, part of my slides is missing, at least on the previous. Yeah, so you know already this bit. So I worked a lot in Leeds before moving to Newcastle, and I have a special interest in high dimensional data analysis, both for new target discovery and biomarkers. So I have worked mostly in cancer and mostly in melanoma, but also in testicular cancer. but nowadays I work in other diseases area as well. So if we take the example of cancer, others have really highlighted how complex it is. So this slide is really to highlight that we need a multidisciplinary approach to deal with this level of complexity, and my own perspective is that data analysis, robust data analysis, lies really at the center of it. Of course, alongside good experimental design and laboratory practice. So. When I talk about robust data analytics, I mean really the way to handle all these type of data. I've been working with most of these, methylation mostly with Miriam as a collaborator. So I don't generate myself any of the data, but I sit right in the middle here. And even animals, etc. Some of these data, they come from very recent evolving technologies. And as a statistician, it's not easy to analyze them if you don't even understand how they are generated. I'm in a group of 35 statisticians, and maybe only 5 of them do work with genetic data. And I have had time to explain them what it is really, what a gene expression is. When I explain it is measured on a relative rather than absolute scale, they run away. So anyway, so we want to improve patients' outcomes via stratified medicine. So we need to find these patients strata, and then we want to design good drugs, and there is a long pipeline to go through, and statistics is involved all the way. So. In melanoma, because that's where I worked mostly, we know already a few oncogenes, BRAF and NRAS, and TET has emerged as well. And the wild type, we call them wild type for these, but they are not wild type for everything. There are other genes as well, which are rarer, which are being discovered. And we know that drugs have been designed for this subtype, the BRAF, so BRAF inhibitors, but we know they don't work always, and there is resistance even when it seems to have worked. And one of the reasons might be that even though it looks like it is one subtype, actually it has itself several other subtypes. And so the hypothesis is if you use different layers of omic, so these are gene expression, this is copy number variants, and you can add methylation and so on. I didn't add this because we didn't have them in our lab. So the idea is we can actually find smaller subtypes, more homogeneous, which can be treated with one drug. So basically, many more drugs have to be found. Hard work for others, not me. So this is the hypothesis. And we know in melanoma, checkpoint inhibitors have transformed really the treatment, and we have seen many countries introducing them, and they started to work. Here we have an anti-PD-L1 survivor. This is ipilimumab, is it? Oh no, this is nivolumab. And this is ipilimumab, yeah. So this is anti-CTLA, this is anti-PD-L1, and then if you put them together. So it is still only 50%, having a good survival after 5 years. So the other 50% don't. So there is still work to do, and so we used the data set we have to try to find biomarkers, focusing on immune responses because of this observation with immune checkpoint. Before going to those analyses, I wanted just to highlight that melanoma is diagnosed more and more early, at early stages. So this is in the UK. Cancer Research UK puts out these figures every year. I didn't have numbers after that, but you can see that in 2013, only stage 1 and 2 diagnoses were 61 and 16. By 2016, it was 66 and 20, so 86% quite early. And we also know that the mortality is the highest, obviously, if you are diagnosed late. It is really imperative to be diagnosed early. However, this is from Australia, where melanoma actually is the most prevalent. In terms of absolute numbers of deaths from melanoma, you have equal numbers at each stage, even from very, very small tumors. The survival rate is great, but because they are a big number, those among them who die quite early are also a problem. They are as numerous as those diagnosed late. So we cannot ignore, biomarkers in early diagnosis, because the number of deaths is also is large. It's only small in proportion, but large in absolute terms. So we need to focus on early stage just as much as late stage. Unfortunately, most of research goes into late stages, understandably, because the deaths in the late stages is high in proportion. So I have been very lucky to be in a group in Leeds for those years, where they collected many cohorts of patients. So I don't have data from animals or from cell lines, but from large cohorts. So these are different cohorts. The largest of them, we call it the LMC, this melanoma cohort. It has been used extensively in GWASs, so using blood SNPs. But then we have also transcriptomes in 700 of these. So this graph, these tables come from a grant application which was unsuccessful. So we were trying to show, look, these are the things we have shown in black. These are the things we aspire to generate, shown in red. And those in gray, we have them in half, part of it. Unfortunately, it was not funded, but you can see what we have already in black. So we have these risk factors in everyone, in all these cohorts. And we have some tumor-related markers and then some host-related markers. By host, I mean from the peripheral blood. So we use this type of data to run our own hypotheses and also to collaborate with others. So in terms of collaboration, just to highlight some of the work we have done in collaboration with others, and you will see Miriam somewhere there. I picked just one. I could have included more. So these data sets have helped us to to advance a little bit in understanding of tumor biology. So my talk will cover these three papers where I used immune profiles, which is known as the immunome, to find 6 immune clusters in those 700 patients using transcriptome, immune markers as a subset of the transcriptome. Refining this approach, Pozniak, who was a PhD student in the group, refined them into 3 using a slightly different approach. I will show you that just in a minute. And then to confuse ourselves a little bit, we also use the whole genome, not the immune markers, the whole genome, after just the usual filtering for quality control. And we found another 6 prognostic clusters, which are different from these 6. And so now we are scratching our head to put all that together to find a unifying framework. So why did we go with immune markers? Just quickly, because for a very long time it has been known that immune infiltrates in melanoma, probably in other cancers as well, but they are quite easy to see in melanoma. They are prognostic. So if you have what they call brisk tumor infiltrating lymphocytes, they look like this. So they really are recruited inside the tumor. Then you have a better survival. If you have none means actually they might be around, but they fail to enter the tumor. There is a kind of a block here. They are stuck, and it is common in melanoma to have some. But if they fail to get inside, then the prognosis is not as good. The intermediate form is called non-brisk. So you have an intermediate outcome. However, even though this is well known, it has not really been introduced in, for example, AGCC staging. You would think they should include it, but the problem is the inter-observer heterogeneity. The histopathologists don't always agree on how to call what they see under the microscope. And you see a lot of patients have these non-brisk because the pathologist has not really found it easy to decide if it is this situation or not, or that situation. A lot of people are put here, about 75 or 80% are put here. So this is unhelpful. So we tried then to find if using immune markers in the transcriptome, you could classify patients according to their immune infiltrates. So that was the aim of our first analysis. So the idea is that if you take a tumor, you remove the mRNA, you profile it, you should be able to spot whether this tumor, people say, okay, this is a tumor gene expression, but tumor is not homogeneous. You want to be able to detect whether inside the tumor there are immune cells. And each immune cell should have its own signature. Obviously, doing it by flow cytometry is an option, but which is very, very expensive. You cannot do it in many patients. Maybe experimentally you can do it in a few, but we want to do it on a large scale. So the only option that is left then is using what we call the deconvolution. So the immune deconvolution we came up with was based on this paper from a group in France, Jérôme Gallon. Some might know that group, and they published this list of genes which are specific to 24 immune cells. So they did extensive experimentation to show that those genes are not seen in tumor. They were using the colon cancer, but we adapted it to melanoma by extensive filtering. And they are not seen in normal cells. They're not seen in the blood. They are not seen in vessels. So if they are specific to immune cell, purified immune cells, you think if you see them in a tumor, then it indicates the presence of those immune cells in the tumor. So that's the hypothesis. So we later then refine this because the immunome compendium from this has also been updated a little bit. So this cohort then of this melanoma, which we used 700 samples. We split the data into two data sets, 2/3 for development and 1/3 for testing. And this training or development cohort, obviously, we did multiple cross-validation as well, as I will explain. Oh yeah, it is here. We used the 300 genes from the genome representing 24 immune cells, and we used the method called consensus k-means clustering in the training data set with a lot of repetitions. So these are called the bootstrap. They are designed to obtain clusters that are robust. If you do a single clustering, there is no evidence that it is real. If you tried it in a different data set, you might come up with different number of clusters. So if you use multiple iteration by using a subset of random samples and random genes each time, and then you average them out, you have robust clusters. And this is what we did. We tried up to 12 clusters, but the data indicated the strongest evidence of 6. And that was done using a consensus, what they call a consensus score. It is like a probability you obtain from those repetitions. And so once you have found those clusters, you still need, so that's a cross-validation, but you still need a validation. So we use this 1/3 left out to validate, to replicate, and a further replication from a completely external data set. So what did we observe? You are familiar with these heat maps, so we have the clusters here. So the training data set and the replication in the Liz melanoma cohort. So the patients are here, and then the genes, the 380 genes, they are actually grouped into 4 bands, as you see. So this band here contains predominantly adaptive immunity, and this band predominantly innate immunity, and you can see it was replicated over the 2 data sets. So cluster 4 lacks both the innate and the adaptive. Cluster 6 seems to have a bit of innate, but no adaptive whatsoever, and cluster 2 has most of both words. So you can see the survivor here in the training, and then when you combine the training and the test set, the survivor is the worst in cluster-4, which lacks both innate and adaptive immunity. And all the other 5 look the same. So it was quite surprising. The consensus cluster analysis suggests there are 6 clusters, but survival, because this was completely unsupervised, survival was not taken into account. Survival itself shows, well, just one of them differs significantly. However, when you adjust clinical covariates, this cluster 6 as well becomes as similar to cluster 4 in terms of survival, which suggests that cluster 6 good survival, you can see it has good survival with all the others, is simply coming from having smaller tumors, younger patients. So using survival itself is misleading here, because the 2 are definitely different in many things. This is what was picked up by the consensus clustering. Just to mention that consensus clustering didn't include covariates. Didn't you have age information or sex, or those things were not present, but they are largely recapitulated in those genes as well. So in terms of looking at covariates, you can see the association of different clusters with a selection of covariates. You can see, importantly, the TILs here. Cluster 2 had most of the immunity, the immune evidence, and about 34% of them have these rare brisk TILs. These are very, very rare to see anywhere else. Cluster 4 had only 7% of brisk. So you can see that clearly the clustering was using an information that is redundant with TILs, as we expected. Those 380 genes were immune genes. And so you can see cluster 6, for example, and I will not go into all the details, but cluster 6 had younger patients, so 51, while others are closer to 60, had less severe disease because no ulceration. Ulceration is a marker of severity. And they were thinner, smaller, and so on. So the survival you see in cluster 6, when you adjust this, cluster 6 has a comparable survival to cluster 4. So this was the first result we obtained with this. And then we replicated that. I'm not showing the replication in TCGA for survival, but you can see in heat maps. So we designed these heat maps to look at a few well-known mechanisms of immune escape to see if in our patients we can see some of them. So these are, in addition to clustering, I created immune score. So I gave to each cell a score, and you can see the selected ones here. Cytotoxic T cell, dendritic cells, and so on. And you can see cluster 4 and 6 have a low of all these. So green is down regulation. And so the second band here, B, shows a host of immune checkpoint markers which have been targeted by drugs. Many in use, others in trials. Originally, we were testing 3 or 4 of these, but then reviewers came with their own list, their favorite. Did you check if this? Did you check of that? Okay, I had never heard for most of these. But when you align them with these clusters and immune cells, you can see clearly they are coordinately upregulated in cluster 2 and downregulated in cluster 4 and 6, with exception of arginase. Arginase 1 and 2. I don't have any explanation myself, but what the take-home message really here was that combined therapy is likely to work better than monotherapy because all these seem to go hand in hand. You can silence one, but the other will do the job. So the next band is showing beta-catenin pathway. You can see that the cluster that lacks immunity has expression of beta-catenin, CTNNB1, and cluster 6 has CMYC. So these two are well-known markers, obviously, of cancer, immune escape. So there was no novelty here, but we were trying to see which mechanisms is playing out in our data. And we ended up focusing a lot on this, and the paper we published actually was, the title was beta-catenin, is predominant in the population, because previous literature has indicated it can be a negligible thing. Yes, it has been seen, but maybe it is negligible. So because our data is population based and it is really representative and it is large enough, we have been able to quantify this mechanism, and we found that 42% of this cluster, no, 60% of this cluster has beta-catenin upregulation, which is limiting the immune infiltration, and it is 30% of the whole cohort. And you can see the same in TCGA. So in the next step, we okay, this is a zoom in actually to show more on PDL1, this band B, to just say in conclusion that combined therapy will probably be better. Are two enough? Maybe not, because we have seen 50% of good response with two drugs. Not yet 3 or 4 or 5. Maybe that's where we are going, but I don't know if it is practical. So the next thing we did is when we found out that beta-catenin is important. This is a zoom in on the band C. A PhD student in the group went down to Sangha to a collaborator actually and knocked down VDR, vitamin D receptor. We know the role of vitamin D in skin biology, etc., and patient survival. And we actually found that there is a relationship between VDR and beta-catenin. So if you knock down VDR, you upregulate beta-catenin. If your VDR is strong, then beta-catenin is inhibited. So that was very interesting to see in the mouse animal. So being a statistician, I didn't do just the score and convince people my score are good. Obviously, you need to say more about them. There are other score of immune infiltration using this deconvolution algorithm. This one gives a total score. It doesn't look at cells individually. My approach look at individual cells, but you can then put together those cells and they have one score. And you can see how well one score I made using this Bindeya compendium correlate with what you get from this other published estimate. And you can see cluster 2 lies there. the highest scores, cluster 4 and 6 the lowest. And when I looked at how the adaptive and the innate immunity correlate, actually the correlation is very high. So the clinician who was head of the group in that time really liked to say it is an orchestra. You cannot rely on one form of immunity over the other. You need both. Again, as a request of the reviewers, we have validated some of these findings with immunochemistry staining because seeing some prognostic markers at mRNA level is not always convincing to everybody. So you can see that some of the markers we showed a good correlation. And this is from TCGA, where they have some protein data as well. So you can compare the protein and the gene expression for a selection of key markers here. Other important observations we did with this data is because it is large and we can afford to do a stratified analysis, we know BRAF is mutated in 50% of melanomas, while NRAS is mutated in 25%, and what type is observed in the remaining quarter. If you look at the survival using selected immune cells like CD8 T cell here, you can see that it doesn't make a difference in NRAS mutated tumors. This is really puzzling, and you can see that it is not by lack of the infiltrations, because NRAS and BRAF seem to have the same amount of infiltration. It is just like they are not activated for some reason. They are activated, sorry, but they are not helping the patient. And NK cell, similarly, they don't do anything really if you have NRAS or BRAF actually. They seem to be only helping patients who don't have any of these mutations. So, as I said, then we refined this signature from six clusters to 3 by a small change in the way we cluster. Instead of using 380 genes, each gene being, a variable. Because I have created those immune scores, then you can reduce the dimension and use only those scores. So, and at the same time, we took the advantage of the Paris group, Jerome Garon, having published a refined immunome compendium. And we reclassified the patient using the new updated immunome and using immune scores rather than genes. So you can see we have far fewer lines here, only a 27 cell score, and then we have patients. And we use the same approach, consensus clustering, using 2/3 for training and 1/3 for testing, and then TCGA for replication. And you can see nicely that the three clusters are separated prognostically. So the 6 we had previously are not separated prognostically, but biologically. And these have an advantage of being sufficiently large. Each of them is sufficiently large for downstream analysis. And we took that advantage and looked at the genes from the whole genome now associated with these clusters. So differential expression. And we found then pathways, and we built these networks of pathways using those differentially expressed. So in a low immune group, the network of differentially expressed genes compared to the other groups is being driven by MYC. So this is a well-known cancer gene, but it was really nice to see that if you do it completely agnostically, algorithmically, this comes on top. It is ranked as the most important with the highest number of connections. And in high immune, if you ask the same question, it is NF-kappa B1 which is given the highest importance. So this was conducted by Joanna Poziniak, who has now moved on over in KUL in Belgium, in Christophe Marin lab. For those who know Christophe Marin, who does a great work in this area as well. So having these groups and having, in addition to gene expression, having copy number variant in tumors, then we wanted to see if there is evidence of alterations, amplification or deletions in these groups. We looked at MYC because MYC can be amplified, and we looked at NF-kappa B1 pathway. Basically, we selected these hits, so MYC and NF-kappa B1. And what Joanna found is that let's start with the low immune. The low immune, we expected the MYC amplification because it was the driver of the network. And then we expected something related to NF-kappa B1 in this high immune. But because NF-kappa B1 is not subjected to amplification that much, so amplification is shown in red and deletion in blue. What happens in the low immune group is actually they don't only get amplification of MYC, they get a deletion of NF-kappa B1 as well. This is NF-kappa B1 is low here, and this is what makes this appear to be high here. It is high in this group because we are comparing it to a group where it has been deleted. And so you can see that each of these markers is on its own is prognostic. And if you happen to be really unlucky enough to have both an amplification of MYC and the deletion of NF-kappa B1, then it is a double whammy. It is the worst of both worlds. But it is quite rare. Only 6 or 8 patients had this thing. So basically, this data have allowed us not to make some incredible discoveries, but to understand a little bit more things that others have talked about, maybe from cell lines, etc., and see how they play out in patients. Because when you want to design drugs for patients, it is good also to know what is the proportion of patients are you targeting. If you end up targeting, 1% of your patients, is it worth it? So these kind of questions. And then we looked at also the association with smoking, because we know smoking reduces your survival if you have cancer. But quite intriguingly, we found that in the whole data, first of all, this is the whole data, smoking reduce your survival, ignoring anything else here. Then if you stratify, you find out in the high immune group, this is the profile in the intermediate and in the low immune. So smoking doesn't seem to make a difference if you are low immune. this is what it is showing, isn't it? So it is intriguing. It can be controversial. So, yep, it was an intriguing observation. Now, as I said, to confuse a little bit ourselves, we use the whole transcriptome now to classify the patient again, the same tumors, and this is by another PhD student. And you can see that using the same approach, the consensus cluster analysis, this time using a tweak of the method, using k-medioids instead of k-means. This allows to be more robust to outliers. So for those who are going to attend my my lectures in next week, maybe I will talk more about that. K-medioids, k-means, and so on. So what this student then found is, if you use the whole genome, your best bet is also 6 clusters. And this time, they all differ prognostically. So the immune markers don't really separate the 6 clusters you find, but the whole genome does. So that was, again, intriguing. This is why we still have to work on it to reconcile all this. But more importantly, what we found is that even when you look at stage 1, we have 200 patients in stage 1, so of the 700. The limiting factor here is the number of deaths. There are only 30 or so deaths in stage 1. So it is a struggle to find the biomarker in the group. But this particular signature shows that a group among the 6 is statistically worse off compared to the others. It is rare to see a group in stage 1 who really shows a difference from the others. So we are still trying to replicate this, because obviously this is a very early observation. But it was interesting that this group 6 has the second worst prognosis overall and the worst in stage 1. Then we use this ROC analysis to try to see if this signature can improve existing, relevant clinical risk factors used to predict survival in patients. And obviously, using stage 1 only because this is where it was the most intriguing. And using AGCC stage is a bit cheating because they are all in stage 1, but there is A and B. So a small minority were in B and the majority in A. So you can ignore this one, but you can compare the signature itself and the known AGCC plus sentinel node biopsy. Because a biopsy is carried out to see the prognosis, to predict the prognosis in a subset of patients, in a few patients, specifically in stage 1. It is more common in advanced stages, but not in stage 1. Those who were lucky enough to have it, you can combine AGCC and the SNB test. You will have an AUC, area under the curve, of 0.74, while our signature have 0.87. So it is better, and then you can combine them and add other things. But this is a little bit, overfitting, obviously, given the small size of the cohort. So the relevant comparison to me is really that one. So as I said, we are still trying to replicate. We are looking for people who have stage 1 gene expression to replicate this signature. The other thing then we did is this class 6 is so important to us. It looks so interesting that we looked at the differential expression of it and the rest. And then the pathway enrichment, and then Rohit Thakur drew this nice network picture. The software used, I think, was Reactom 5As and Cytoscape and Santiscape, these type of tools. And you can see that it is driven by CJUN. So if you ask the most important driver here, it is CJUN. And CJUN is a well-known marker of the EMT, and it is also correlated with AXL, which is known to be a marker of response to immunotherapy. So that was really interesting. We went into literature to find data set where they might have used immune checkpoint blockade and generated the gene expression, because we didn't have a treatment. I didn't say it, but our patients are treatment naive. So the recruitment started in 2000, where there was no treatment apart from removing the tumor and following up the patient, really. Some of them, when the disease recur, it might have happened when new drugs have arrived, and then they were put on them. But the gene expression were generated at a time where there was nothing. So in the cohort, a couple of cohorts here, one by Hugo et al. Published, where 27 patients were treated by either nivolumab or ipilimumab, or actually it was all nivolumab. We had a number of responders and non-responders, and you can see that the non-responders were predominantly in our class 6. Okay, and then we did the same in another cohort. This is where all 6 were non-responders. So it was intriguing, and as I said, we need to replicate this signature. So my latest work has tried to look at these immune subtypes. So the melanoma subtypes, which they are generated using gene expression, to ask whether you could see them using histopathology images, because these are available in many hospitals. They are probably underused. If advanced modeling tools could interrogate them and maybe find the same groups as those we found using gene expression. Maybe in the future, you might not need gene expression anymore because it is expensive and takes time. By going straight into the analysis of those histopathology images, you can find the same thing as the gene expression would have shown you. So another PhD student then in Liz, who is actually writing up and will finish in a couple of months, has worked using neural networks to analyze these histopathology images using some very complex deep learning and try to predict just the immune groups so far, and separately to try to simply predict survival. So because immune groups are prognostic, if you can predict them, that's good. But also, ignoring immune groups, you can ask if these images can predict survival. So she's done that, and you can see using a whole lot of different methods. MIR and its variants. So MIR is really very, very important in image analysis these days. And you can see the kind of AUC we are getting at. some of these are very good with, you know, up to 80%. So this will be my last slide, I think. Yeah, in a summary, so these data, we have really extensive molecular and clinical data, and we use them to learn a lot of things that are going on in the patients, and then those things can be tested in the lab, or the other way around. People can work in the lab and then make some hypotheses and then ask themselves. Very commonly, the reviewers will wonder, how does it work in patients? And so we are really very happy to have been involved in collaborations to help answer these questions. And so my group tries to put together methods that can help for these type of questions, and remains always open to collaborations. Thank you, and I think we have time maybe for a few questions. Hey, last slide for acknowledgement. So Miriam, of course, and the PhD student who did all the work as always, and many people. I worked in a lab led by these two people before they retired two years ago. Then I inherited all this data, which I helped collect, but they were the clinicians, so they played a bigger role in collecting them, but they are mine now. So yeah, thank you.
Thank you, Jeremy. Questions?
Thank you, Jeremy. Very interesting analysis of melanoma data. I was just curious about the data that we currently have, and if you're generating new data based on the new therapies that are being used for treating those patients, and how could they compare with those treatment-naive patients that you collected before?
So, as I said, our patients have not been treated for a long time. If they have, it is like 8-10 years, because the follow-up is up to 15 now, or even 20 for some of them. So we don't think the treatment has affected their survival, because it came, like 10 years after they survived already. The question maybe is why the relapse happened so long after. I think that's the £1 million question. If you have data set where treatment is known to be given, obviously it is interesting to run new analysis. We are looking, the hypothesis is that because these immune groups correlate well with, seem to be related to the targets that are being targeted really to treat, PD-L1, PD-1, CTLA-4. We think that if you have this immune gene already, you are predisposed to respond. So we are looking really the way to collaborate with people who have treatment to see if these groups are predictive of response or not, or if new groups need to be found for those treatments.
Questions?
Congratulations for the lecture. I'd like to know if the profiles, these molecular profiles of these 700 about tumors that you showed, are they RNA-seq?
So it is microarrays. So they are old arrays that preceded RNA-seq, but they are no different to RNA-seq, because TCGA, which is RNA-seq, we always validate with their data. So the analysis is slightly different. You need a lot of data cleaning. Basically, that's why they moved from that platform to RNA-seq.
Okay.
But they are similar.
Yeah. Okay. So, but some genes are not represented. So, if we wanted to correlate to some long non-coding RNA or so, maybe they are not represented.
We have many of them because Miriam and Attilas, we have been able to work together and find. From my from memory, there are 500 meals. I think Frank said that 2,500 meals in the genome. This array has 500.
Okay. I think so. The other thing is when you show the data about a vaccine to MAGE-3, that means it was actually done in humans, these vaccines, or these are in mice? I didn't quite understand.
I don't think I talked about
it. Was MAGE-3 antigen vaccine, and then
which slide is it?
There was by the end of the talk, by the very end of the talk, and then the response. There before.
The next.
The next one, yeah.
This one, okay. So these are the cohorts. These are cohorts available to download really from public repositories. So I believe it was anti-PDA PD-L1. And this is a MAGE-3 antigen vaccine, which is also an immunotherapy, just to show that this group also correlate with bad response to this vaccine. So it is not my data set.
But it means that if you combine the treatment with the vaccine, the responders are worse. I mean, the response.
So in this data set, which is available from public repository, what we did is to download it and classify the patients. There were 55 patients. Classify them into our 6 groups. Because it is gene expression, you can do that. And then look at the response they reported. They were treated with this, and we have no control on that. So I don't know if they were treated with a combined therapy. The hypothesis for me is that the response will be higher because our data set shows no, this is not checkpoint blockade, though.
Yes, so I understood now.
In the checkpoint blockade, it is not enough to block one, but for a vaccine is something different.
Yes, I get it now. At the beginning, when you showed, I thought it was a combination.
No,
thanks. Yeah.
No, that's two data sets.
It's okay. Thank you, Jeremy.
You're welcome.
Thank you. So now we are reached the end of this wonderful meeting. I'd like to thank you very much, all of you, especially our inviter researchers. Thank you. If someone, if you want to talk with them, you can let me know. Then I can schedule a meeting or something. Please feel free. Thank you.